And feel free to utilize the chat box. Feel free to unmute.
I see, this is Juanita. I see several things. I see teamwork. I see the efforts to put together all the nuts and bolts that needs to make whatever this is run smoothly. I see hard work and efforts.
Absolutely, absolutely. And Tanya in the chat box says a similarly collaboration. Is there one member of this team doing, like, whose job looks more important? Is there a particular individual whose job looks more important? What about the gentleman on the top right? He's pushing a pretty huge object. Is his job more important?
I don't know about it being more important, but I think I see it because it seems like he starts the process. And how he guides this instrument determines the direction it's going to go in. So I don't know if you want to say important, more important, or it's valuable or leadership, because he has to guide the team. Seems like, because if it doesn't head in the right direction, things can be pretty messed up. It seems like.
Absolutely, and I see in the chat, Raphael mentions everyone must put in the same work and effort in order for the whole system to work. Omar stated equal. Robert, some is more precision, some is more strength. So absolutely, I think the important point is in order for a true team to work the most optimally, everyone's role has to be recognized, and everyone's role is important.
Well, Kiosha, this is Miss Plus. Hi. When I first looked at this on the right bottom, it looked to me like that person is trying to bring that back into the group, because it looked like it got off by itself.
Yeah, definitely.
And so he's trying to pull it back in, and the one on the left with and I don't know if that's after he did or whatever, it seems like to me that he's trying to get it back in the groove.
Definitely. So just making sure that all the pieces are together, right? There needs to be at least, one person just making sure that nothing is getting away, right? That everyone is coming together and all the pieces are in place.
And I think looking at the bodies of each one, looks like they're doing whatever they can to get it back all together. Sometimes you have to go an extra mile to be sure that everything fits in, instead of just, but it looks like they're really trying to collaborate together and pull it together.
Absolutely.
If I could add real quick.
Sure, absolutely. Let me go back.
So I actually see a lot of opportunity. I see this as a very beginnings of a plan or a process. And the opportunity that I see is in between some of the cogs, there is space for pigs. So it could become a permanent system. So I see this as a very drafty process, and definitely an opportunity for these people to sit back and actually look at the progress that they've made by creating this machine with pegs, so that these cogs are actually held in place permanently.
I love this. Yes. Excellent, guys. Other thoughts? Okay, so what exactly is team science? We hear a lot about teams, the importance of team work, but there actually is a science with certainly ever-growing research in this area. So team science, according to Bennett and colleagues, is a collaborative and cross-disciplinary approach to scientific inquiry that draws researchers who otherwise would work independently or as co-investigators on smaller scale projects into collaborative centers and groups. Okay, so it's certainly a science that emphasizes working together to to solve problems, to push the knowledge base forward in a respective field. And it's nice to look at a research team as a continuum. So on the far left has a low level of interaction and integration. And so this is often thought of like as a traditional model where you have the independent investigator working by his or herself on a research problem within his or her laboratory or own space. And then the continuum moves farther to the right where there's an integrated research team, and recognizing that each research team has specific skill sets, has specific knowledge, experience, lived experience that they're bringing to the table to address the research problem. In a fully integrated research team, there's structure, the teams meet regularly, the teams agree on goals, objectives, next steps, leadership responsibilities are shared, decision-making. And it's really thinking similarly to the figure that we just discussed, right? Being very, very integrated and everyone having an integral role. So why should you care about team science? These are, some of the areas that we felt are certainly important to think about when you think about the importance of team science, and certainly jump in if there's something that should be here that you feel should be here that is not. But increased productivity. The research certainly supports this, that teams are more productive. Teams that are structured most efficiently certainly are more productive than individual traditional models. It may be required for a grant more and more. And this is important. I mean, when you think about, various ideas, various, issues, various problems that should be examined more closely, it's important to have folks who represent different areas, different sectors, different disciplines to come together. Knowledge production, it's important to facilitate knowledge production. It's important to build scientific and technical human capital. And also, because you're having different folks coming together, it's certainly an opportunity to embed cultural and context to whatever you're studying. This is particularly relevant to, for example, substance abuse, right? Substance abuse research, where that context is going to be just as important. And it's also fun. I love working in teams. Ms. Norma mentioned a broader perspective in looking at questions. Absolutely, that's a fantastic point. Any other additions? Dr. Friedman, do you want to talk about this one?
Yeah, thank you, Kiosha. Great job so far. Many grants are emphasizing team science. There was a white paper in around 2005 that suggests the NIH moved to funding more multiple PI projects, and this multiple PI program director option started by the NIH in 2006. It's already been 14 years. When you write a grant and you have a multiple PI or program director, you have to justify why it's being used, how it's going to be used, whose responsibilities are what, and what the roles of the different people are. So for many grants, this is the new way to do it. And I think most grants that I review and I'm part of have this multiple PIPD option. Some grants, however, there's a grant that we are eligible at Charles Drew called the SCORE grants, the SE1 grant, for example, part of that. And also a lot of K awards really stress career development. They don't allow co-investigators. So they often have a mentor, but usually not a co-investigator. So you have to look at the grant mechanism to see the options. Next slide. And then this I got basically from the NIH page. There are different type of collaborators. The first one that I mentioned in the last slide is the project that has a multiple PI or program director. Both of them are equal. The next option you can have is one where you have a PI, principal investigator, and they have a mentor. You can have a co-investigator. A co-investigator is an individual involved with the program director, principal investigator, in the scientific development or execution of the project. The co-investigator, the collaborator, may be employed by or affiliated with the institution, or maybe under another institution. The co-investigator typically devotes a specific percentage of time to the project and is considered a senior or key personnel. A consultant is somebody who usually is not as part of the institution and provides professional advice or services for a fee, but is not employed by the institution. And finally, you have community collaborators. These are very helpful to get a community perspective on the grant to help with information and dissemination of the grant.
So many of you may, I'm sure have experience participating on a team, whether it's, in research or outside of research. But one of the things that we urge you to think about really as you approach new team is, am I ready to participate on a research team? I think, it's important to think about what you're bringing to the table as well, in addition to your values, experiences that you've had, your personality type, situations that work for you, and not so much. Really, working on teams offers an ability to grow. But before you do that, it's important to self-reflect, engage in an active engagement of self-reflection and exploration, asking yourself, what is your communication style? What is your optimal communication style? This is extremely important, especially in this in this virtual environment, right? That we're all working more in. Thinking about, I know Zoom is very, very, very much draining, right? But, perhaps emailing, picking up a phone, thinking about what's your optimal communication style? What is your approach for managing conflict? What is your personality type? And how do you give and receive feedback? Do you give and receive? Do you ever give feedback? When you receive feedback, are you open to it? And then also biases. This is so important. We all have them, right? It's important to recognize them though, and think about your biases and really checking them at the door. So in terms of personality type, many of you may be familiar with the Myers-Briggs assessment, Myers-Briggs assessment. And I highly encourage you to complete the assessment if you haven't already done so. Here's a website, but there are other assessments available online that you can certainly take advantage of. Also asking yourself, again, in terms of how you operate, how collaborative are you? Working in a team, is maybe more effort for some than others, especially if you are used to, kind of just depending on your own self, right? Depending on your own work, maybe working with one other person. When you're working with a larger group, it's important for you to let your guard down, be open to collaboration. How argumentative am I, right? Understanding that you may not always be right, okay? Your opinions definitely matter. However, it's important to be considerate of others that you're working with as well. Everyone's thoughts and opinions matter as well. So again, you're working together as a team. So there are certainly fundamental elements for effective teams. Hall and colleagues did a great job kind of summarizing the characteristics of an effective team. We're not going to go over each characteristic, but just ones that we really want to touch on that stand out to us based on our experience. But here are some listed. The first I'm going to go over is trust. Because really that's an important area for any team. You're not going to work most effectively if you do not trust one another. And so working on an efficient, well-running, smooth team requires a mutual dependence. And there are different types of trust. There's identity-based trust, where members of the party, members of your team really understand and appreciate each other's wants, desires, and values. There's a calculus-based trust where individuals on the team interact with each other, and everyone understands that, you know, deadlines, ensuring that everyone keeps their word. This is where communication is extremely important, but everyone kind of runs most efficiently because folks are doing what they say that they're going to do. Competence-based, built on the confidence that we have in the skills and capabilities of one another. And then swift trust is another area. It's built on ensuring, just giving all of the members of your team the benefit of the doubt that, you know, regardless, everyone has good intentions. We know that trust has an impact on each team member's judgments about one another's abilities, about their observations, designs, and ultimately scientific results. When you're working together on a team, everyone is contributing, right? Is contributing their own piece to the puzzle. And if you're doubting what a team member's contributing, then that's certainly going to impact how you feel the results are headed, how you feel the findings are headed. So we're gonna pause and go into an activity. And one question that I want you all to answer, and you can please use the chat box for this, is what is unique about you? What is most unique about you? Go ahead and type it in the chat box. I'll give you about a minute to think about it. I know it's at the forefront of everyone's mind how unique you are. But go ahead and think about it and enter it into the chat box. These are great. Okay. Few more seconds. These are terrific. Great. If you haven't had a chance to enter what's unique about you, please do so now. These are awesome. Okay. Very unique group indeed. So who would like to share what they typed in the chat box? Go ahead and unmute yourself and state what's unique about you.
Hi, everyone. Can you hear me?
Yes, I can. Yes, we can.
My name is Tanya Lugo, and I think that something that makes me unique is my ability to be resilient in different situations. Especially when it comes to research, I know that when I conduct experiments, there's a lot of failure in that, and the ability to bounce back and look at different solutions or even techniques to achieve my goal. I think that's what makes me unique.
Thanks for sharing, Tanya.
Hi, can I go next?
Absolutely. Hi, Hannah.
Hi, my name's Hannah Pons, and I wrote that I'm dedicated and I strive for greatness in everything I do. As an athlete, I've been an athlete for 15 years, so I feel like that gave me a great background and perspective on life to keep going in whatever you do. And just strive for greatness, because you never know what could happen. You never know what you could achieve if you keep going.
Absolutely. Thanks, Hannah, for sharing.
I'll share. My name is Professor Oxala Dillard. I think what makes me unique, or what has first come to mind, is just my acknowledgement of my intersectionality in the world. And so I'm a member of multiple identities, and I think that really lends to my understanding of research, different research populations, groups, as well as working among a team. I'm also a young researcher, so I think that helps as well, kind of shifting some of the paradigms of research and ideologies of research. And I'm open to unique and fresh ideas.
Thank you, Professor Dillard, for sharing.
Hi, everyone. My name is, sorry.
Oh, no worries. Go for it, Robert.
My name is Robert, and I've been really lucky in that I've been able to facilitate a lot of speaking groups. I've also, when I was younger, I was a peer counselor, and so I feel like I carry a lot of people's stories with me. And so when there's an issue or a problem that we're trying to solve in research, I pull from a lot of that, people's perspectives, people's stories, and trying to kind of fill in the holes wherever we're missing insight.
Great. Thank you. Thank you. So this was great. Excuse me. Just we didn't. I definitely, if you haven't already done so, excuse me, glance through the chats, read about the uniqueness in folks on this Zoom call. It's pretty great, pretty amazing. And, you know, when you think about a team, it's important for you to have the ability to connect with one another. And so activities such as this is great for that. I'm hoping that as you're hearing others, as you're reading about the uniqueness in others, you're building connections in that, like, wow, you know, I too am resilient, right? I too understand that I represent various backgrounds, right? And I understand intersectionality. Okay, so those are that connection is critical, and there's also it's also important that that you're vulnerable, that you show vulnerability as a team member as well, because that is going to facilitate trust, right? That you're human as well, and many of you are leaders and are going to be leaders. And vulnerability is an extremely important quality in a leader, because it's going to facilitate trust even more quicker among team members. And it's important to proactively build trust. So not just wait. Ever since the starting with the inception of the team, it's important to communicate, set expectations, understand what your outcomes are going to be, how data is going to be shared, what is the role of each team member, develop and implement any procedures, ensuring that you're all held accountable, and what will be the mechanisms for ensuring accountability. And most importantly, building trust takes work. It's not something that's going to occur overnight. It's going to evolve over time. So another aspect of building a team includes developing a shared vision. And certainly this comes with effective leadership. Sometimes, especially in the research environment, you might have someone come and, you know, bring everyone together and say, hey, I have this amazing idea. You know, I know you guys' skill set. Let's meet to discuss, right? And so it might start with an idea of the leader of a team member, but it's going to also take further discussion, further articulation, right? So that everyone has ownership and everyone comes to an agreement. It's important that the roles and the responsibilities of each team member are in alignment with this vision. It's important that the resources are in alignment with the vision, that the team members, every team member, understand what resources are available as well. And also, again, ownership is so important. So each team member should really have a say in terms of how their research goals. Each team member should really understand how their research goals really are in alignment with the vision. Also, in work, I'm sorry.
Yes, I wanted to add the comment that I put in
Yes.
here. I'm a big fan of the Franklin Covey principles. They have 7 principles of highly successful managers. One of them is have the end in mind. So I think I try to live by that precept. Try to think what your long-term goals are and get focus on your long-term goal, not just little daily ups and downs.
Absolutely. Strategic identification of team members is also critical, right? You know, the phrase "you are only as strong as your weakest link." It's important for you to really be thoughtful about the team members, um, uh, ensuring that team members all have a sense of commitment and dedication to the project at hand. Really recognizing the strengths and abilities of each team member and thinking about how to best optimize each team member's strengths and abilities. What is in it for me? I think you know, it's important for you to ask that question. It's not a selfish thought. I mean, it's important certainly that you think about the overall team, but we're talking about research, right? And so perhaps again, you have a specific question. You have something that you can carve out, right? Especially if you are a junior researcher, right? Perhaps there is a publication that you want to ensure that you aim for. Perhaps there is a grant opportunity that you can strive for as well. So really thinking about your career and what is the end goal. What do you like or dislike working with? Mentorship is also important, especially for you know, any junior folks in the team. It's important that you have mentors on the team, and often like the senior person. This is why studies show that great teams have a nice mixture of definitely a senior person, at least a senior person, some mid-level junior folks as well. Strategic identification again. So these are just some questions to help guide you. I'm not going to spend too much time, so I want to have time to go to other slides, but these are just some additional questions as well. Promoting disagreement while containing conflict. You know, you're not always going to agree on everything, and that's fine. But what are the steps that should be taken if there is disagreement, if there is conflict, and you know, how is it going to be resolved? Is it should mediation be a part of this? Should there be a governance committee? You know, oftentimes when you are when you're working on a team, it's important to note that disagreement and healthy disagreement is what's going to help, you know, push the group further, right? In terms of the seeking of knowledge. So perhaps you see things one way, and there's another member on the team that, you know, completely doesn't agree. And so it's the entire team should be very open to this disagreement. And again, as long as it's moving the work forward, everyone on the team should be open to giving and receiving constructive feedback. There should be clear expectations for sharing credit and authorship. And this is something that, that should be established early on, not later on. Just a simple things such as authorship criteria. Presentations, right? It's important that everybody on the team knows that members on the team should be identified, should be acknowledged, appropriately attributing all people who contribute to writing, who performed various experiments, provided any intellectual input. And diversity should be harnessed. This is true, this is important. And, you know, I have here, when folks think of diversity, don't just think about race, right? Don't just think about ethnicity. There are other areas, right? There's gender, there's sexual orientation, there's gender identity, there's title, there's ties to the community, discipline, age, Robert, thank you, years of experience, education. There are many elements to think about, but diversity is indeed a strength. It helps to facilitate trust as well. It's important that the team appreciates and respects all perspectives. And again, everyone has something valuable to contribute. Okay, so we have a few case studies that we thought we would discuss. Would anyone like to volunteer to read this case study?
I'll read it out loud. This is Plus.
Thank you, Ms. Clark.
Dr. Bello and colleagues have been sharing ideas for many months around a possible new project. Some preliminary data from one of the team members resulted in a lot of excitement. The group started to coalesce around what felt like a promising direction for a new research project at the intersection of several disciplines. As the team became more excited about the project and generated enough data to submit a grant application, Dr. Bello started integrating time into their meeting agendas to ensure that the dynamics of the team were working well. Dr. Bello had extremely strong self-awareness as a result of taking advantage of professional and leadership development opportunities at work. Dr. Bello was able to help the group develop trust, surface tensions early, and often, and openly challenge and question research results. In addition, team members took time to create a research plan that outlined roles and responsibilities and talked about how they would hold each other accountable to to the commitments each of them made.
Thank you, Ms. Bliss.
You're welcome.
So what made this scenario successful? Feel free to use the chat box or unmute. Okay, so Dr. Bello, thank you, Robert. Dr. Bello had the success of everyone in mind. Anna mentioned early communication, okay? Raphael, I mentioned communication amongst the colleagues, absolutely. So communication.
Dr. Bello was open enough to accept everyone's opinions, ideals, thoughts, and that made the team more cohesively and they worked together.
Absolutely.
And they built the trust.
Trust, communication, absolutely.
Mm-hmm.
So who would like to volunteer to read this one?
Can you hear me, Kiosha?
I can,
Okay, this is Juanita. Okay, I'll do it for now. And I also want to say I'm going into the mountains and I may lose reception, so I'm going to do this now. And I'll log back in when I can if I lose. But I'm going to read this.
Okay.
So team members on an interdisciplinary multi-institutional research project.
Okay, I think she might be losing reception. Miss Juanita? Oh, I can go ahead.
You want me to read it?
Sure,
I can read
thank
it.
you.
Okay.
And
Team members are
mute her.
Juanita, your phone is messing up really bad. Okay, team members on an interdisciplinary multi-institutional research project established a publication and data analysis committee. This committee was charged with ensuring the team adhered to the International Committee of Medical Journal Editors, ICMJE,
Yes.
fair authorship guidelines. It also provided a forum in which decisions on authorship and related issues were openly made by an assembly supported by all investigators. This committee was also empowered to review and approve data analysis plans and study-wide papers. The processes for submission were clearly defined by the committee. Over the course of several years, all issues that came before the committee were handled satisfactorily.
So what made this scenario successful?
I forgot what I just read.
Let me go back. Pre-planning, yes, yes, absolutely. Thank you, Ms. Norma. Pre-planning. Okay. So we're gonna move forward. There is, let me see. We have about 5 minutes. So there are other scenarios, and these slides are going to be made available to you. There are other scenarios that are not, that demonstrate the team not working as efficiently. So we encourage you to take a look at these and think and ask yourself, what were some of the things that did not go quite well? Okay, so I'm going to skip through these really quickly. There are institutional and organizational influences that are going to impact your team. Some things to keep in mind. For example, funding, and then also institutional structures and policies that may be more or less supportive of your team. So it's certainly something to keep in mind. And then also team size. This is, I think a lot of folks are like, what is the most optimal size? Is there an optimal size? There is literature that supports the association between large research teams and research outcomes, in addition to research productivity and impact. But there is, I did come across some data that shows that there are smaller teams that are able to kind of get a little more deep into the weeds. They might not be as productive, but in terms of, of actual findings, their questions, they might be able to probe a little more deeply. But ultimately, I think certainly large team sizes are effective. In terms of opportunities, technology is an important opportunity, especially now, right? It's allowed us to utilize various platforms to increase engagement among team members. We utilize various platforms here at CDU, and I'd be happy to hear about some of the platforms that you all are using that assists you in terms of your team. Also, community partnership and engagement. I cannot stress this enough. This is an extremely important approach and an opportunity to partner with community, understanding that that lived experiences is just as important as evidence-based, right? It's important to merge the two, especially with many research going on today, and also multiple sectors. And then there are additional opportunities as well to work with others. Challenges, so I mentioned funding, that is a huge challenge. Education and training in team science, it's important that there is more training opportunities, that team science be incentivized often, and this is not just in the funding realm, but it's also institutionally, right? When we think about, for example, promotion and in an academic setting, our teams and working with teams, is that really, that's not really that factored in as much. It's all about, your publications. However, we know that in working with teams, you probably are going to be more productive with your publications. And then time restraints. Oftentimes, it's difficult to assign time to work effectively in teams. So Dr. Friedman, do you want to talk about?
Yes. So just do Kiyoshi, just do them one at a time, maybe. Hopefully.
Oh, sure. Sorry.
Okay. So this is an assignment. We don't have too much time, but people can think of a project they want to work on. Write that down. You put it in the chat first of all, just real briefly. Sickle cell disease. Okay, go to the next one, Kiyoshi.
Okay.
Think of who's currently on your team. Okay.
What's the next one?
Go to the next one, yeah. Think of gaps in your knowledge of technology, your current team. So you have people that are good in certain areas, but you're missing a certain area. Think of those gaps. You know, some people have gaps in statistics. Some people may have a gap in clinical trials. Some basic science people may have a gap in certain technology they need to add to their team. Think of who you can add to strengthen your team. So you identify these gaps. Who do you know on your colleagues, on people on campus, people in other parts of, you know, now with Zoom and technology, we can have, you know, people around the country basically can help you with your team. So think of who you can add. Raphael says electronic nicotine effects, its effect on the vascular system. Excellent. And strategize on how to approach them. So, you know, I think one of the key things is really to try to incentivize your team member to join your team. To try to tell him or her the pluses and minuses of being on the team, how you're going to meet, when are you going to meet, what's in it for them, and as some of the things that Kiosha said you want to talk about early about authorship, grants, future directions. Does anybody want to chime in on some of their thoughts on this assignment and how to build a team?
Hi, this is Norma. Can I chime in regarding your invitation and selection? I've got an echo here. Selection of community partners and keeping in mind the strengths and resources that they may bring to the table. And I have to bring up the long-term partnership I've had with Dr. Friedman since about 2013. And I've been able to do so many things that you wouldn't think of initially when you think of the community partner coming on board. Because of my background, I bring a wealth of administrative experience in terms of having been able to help with the budget, having been able to help with recruitment and you know, reviewing resumes and whatnot. But just remember that the community, all they've done to stay alive was write grants. So remember that there's some other strengths and resources that come along with the community team members, other than just helping to recruit for the study. And a very novel thing that's happening at Drew that I'm sure you're well of is the major basic team science that we've had going on for a long time. And yes, I still say we because I'm still part of the team.
Yeah, Norma's been a fantastic colleague, team member, great friend, partner over these years. I don't think I could have succeeded without her, and I think we work well together. And I think having somebody in the community, as Norma said, it really adds to your perspective on the tackling the issue. And as I said earlier, think about who you like to work with, who you don't like to work with. I love working with Norma. It's a delight, and there's other people that I'm not going to mention, but some people I wouldn't want to work with. But when you build your team, think of somebody who's gets along well with you, easy to get along with, things like that. Anyone else want to talk about teams? Okay.
This is Plus again. I just want there might be some on the call that might not be aware of the community faculty on campus. I'm now working with Dr. Friedman. I've worked with Kiosha and interviewing and hiring and whatever, and Dr. Martins and whatever. But sometime, like you said earlier, lived experience is a lot of value to whatever you're doing. I like to work with people. Some people like to work with animals. I'm not the animal person for research, but my specialty is mental health. And so, and I'm not ashamed to talk about it and bring it to life. So we have some faculty, community faculty that do all kinds they bring all kind of specialties to the table. And so you might reach out to them. And two of my mentees are on the call. And so one is a surgeon and one is, I can't even think what she does now. But anyway, you know, I'm willing to learn what they do. And so it'll be a win-win.
Thank you, Ms. West.
Yeah. Thank you.
Thank you.
Any other thoughts, comments?
Okay, you have your
So this concludes our presentation. I don't know if there are any additional questions, but thank you very much for your time, and good luck to you all. And again, we'll definitely see you soon.
Thank you, Dr. Partlow. Excellent.
Thank you, Dr. Partlow, and thank you, Dr. Friedman. Those were wonderful. And next we have Dr. Martin Lee. Dr. Friedman, would you like to introduce him? And then after Dr. Lee's presentation, we will have a talk.
So Dr. Lee is also one of my favorite team members. I've worked with him for a decade or so at least, and he's also delightful to work with. We talk about teams. Dr. Lee is just somebody that's just very responsible, professional, gets back to me immediately, even if he's traveling all around the world. So he's a delightful team member. He's a professor of biostatistics and CTSI service statistician with expertise in pharmacological statistics and clinical trials. He's on the faculty at Charles Drew, teaches statistics at Charles Drew, and collaborates with many investigators, especially in the general area of epidemiology and diabetes. Dr. Lee collaborates with the VA Greater LA Healthcare System in several roles. He's a CTSI investigator. Dr. Lee works on statistics, collaborates with people in the CTSI, and helps with training in areas related to statistics. So we really thank Dr. Lee for being part of our Research Institute today. Go ahead, Dr. Lee.
Okay, thanks, Ted. I appreciate that. I just need my slides up. If somebody can put those up for me.
Do you have your slides on your computer that you want to just share?
Okay, I'll have to just give me a second. I thought.
Or Christina, do you want to put us up?
I got them right here.
Okay. Then you can control them.
All right.
Perfect.
All right. Okay, can everybody see those?
Yeah, looks good. Thank you.
Okay. Well, thanks a lot, and I appreciate that. And boy, we're really changing direction after that that really nice presentation by Dr. Partlow. I hope to edify everybody on the right way to pick a statistical procedure. But before I start, I just wanted to make a couple of comments. I've also, thank you for the nice introduction, Ted, but I also wanted to point out that I'm at UCLA. I've been a professor there. I'm starting my 42nd year. And when I always mention that to my students, they look aghast and I go, yes, I was actually teaching when I was 5 years old. So in any event, the other point I wanted to, the other question I often get when I first talk to students in particular, and that is, what exactly is a statistician and what do they do all day long? And I tell a quick little story that maybe elucidates this. I went to school with a friend of mine, a colleague of mine, way back in the day, and we went on directions. I ended up in clinical research and drug development and that sort of thing. And my friend decided to join the CIA, and you know, and that makes sense because there's a lot of analytical work that's done in decoding information. But he actually went out into the field and went into liked to work in very dangerous areas, and one time, unfortunately, he got captured by some particularly unsavory people, and they wanted to know what he knew. And so what happened, unfortunately, and I know this doesn't sound great, but they put him to the test, and they wanted him to give every information, any information he had. So they brought him into a room, they strapped him to a chair, and they gave him 2 buckets of water, one that was freezing, absolutely freezing cold, the other one that was boiling hot. And they stuck one of his hands in the freezing cold water and one of his hands in the boiling hot water. And he started to smile, and his captors asked, "Why are you smiling?" And he goes, "Because on the average, it feels great." So that's what a statistician does. And by the way, that's just a joke, not a real story. Anyway, so let's talk about choosing the right statistical procedure. Whoops. And so what we're going to do here, and I'm assuming that that the audience knows a rudimentary amount about statistics, but I'm not going to assume very much. And so what I'm going to talk about here today is how we organize statistical tests, some of the definitions associated with statistical procedures, different types of measurement, which, by the way, as you'll see during this lecture, becomes the key determinant of what statistic you use or what analytical test you use. And this is a point that I make early on in my class, usually in the first lecture or so, that it's really important to figure out what kind of data you either intend to collect or you have in front of you if somebody's given you data. And that will really drive the statistical approach that you're going to use. We're also going to talk about how descriptive statistics are almost as important as the actual analysis itself. And then we'll take a look at a couple of flow charts, one in particular that really kind of combines everything in maybe the first year of beginning statistics into one picture, which is kind of amazing when you think about it. So let's first of all talk about designing a research project. We have empirical questions. In other words, what do we want to know? And I give an example of a study that was done in 2008 by Tross and colleagues, in which they're trying to show that by practicing safer sex, women practicing safer sex, who prevent the development or the transmission of HIV and sexual transmitted disease in women who are in substance abuse programs. And so what we're really talking about here is then the statistical questions in analyzing that kind of data. And so here's a neat little diagram that comes out of a textbook from a few years ago. So in science, and one of the things that we need to remember is that statistics is science. I like to point out that about 100 years ago, statistics was invented as a discipline in order to put science on a strong quantitative footing. And what does science do? Well, what science does is it thinks about the world, a population, if you will, or a universe, if you will. And it doesn't matter what type of scientist you are. If you're an astrophysicist, the world is literally everything. If you're a particle physicist, the world is what goes on inside the atom. But nonetheless, the world is what you're trying to decide. And then you have a theory about whatever your discipline is. And obviously, in the kind of research we're talking about here, you might have a theory that a certain type of program may benefit people who are suffering with substance abuse. And so, obviously, once you have that theory, you want to you want to test that hypothesis, or that theory gets transferred into a empirical hypothesis, and then you collect data. And as you collect data, and then you analyze it, which is what we're going to talk about today, that of course revises the theory, and then you go through the whole cycle again. And if you think about it, that's science. That's what the scientific method is. That's what we all learned how to do when we were first learning about science many years ago. Now, in statistics, we have a number of different things, words, terms that you'll see identified or talked about over and over and over again. The first thing that I like to mention is what we call descriptive statistics. Excuse me, I have hard time saying that sometimes. Descriptive statistics, which are basically the first thing you should always do when you get data. Look at your data. You know, like I said, tell my students, get dirty with it, roll in the mud with it, see what it looks like. Because without a careful understanding of the data, you can overlook things like problems with the data, outliers, unusual observations, seeing things that otherwise you might not have seen if you just go into the analysis, and then the analysis may not make sense. And so really what we're talking about here is just summarizing, organizing, and simplifying the data. Then, of course, we jump into the other and obviously broader category of what we do statistically, which is what we call inferential statistics, which are the techniques that from where we infer, we draw conclusions from the sample to the population, whatever that population is, whatever we choose to define it. Now, one of the things we understand or learn about basic statistics, first and foremost, is that anything that we come up with statistics is not going to be exactly right. I know it comes as a shock sometimes for people to say, well, you're dealing with math here. Math is always right. And that's the interesting thing about statistics, because we only get to see a small fraction of what we're interested in. If we're interested in studying people with cocaine addiction, for instance, we only get to see that sample that's in our study. We don't get to see everyone in that universe. And as a result, the conclusions we draw have error associated with it. Now, the word error is an unfortunate term, but it's what we use in statistics. But it's not truly the kind of error, you know, like we normally think of that, which is like a blunder or a mistake or some kind of problem that we could rectify. We can't rectify what we call sampling error. It's the difference between what we observe, the sample, and what is the truth, that is the population. And we know for a fact that there is a difference. Now, you hear about sampling error all the time, but may not recognize it as such. For example, we're hearing pretty much on an hourly basis about another poll or in the election. And the first thing after they mention the results, you hear another term, margin of error, same idea. That means that the results that you're seeing are, there's a possibility of a discrepancy. In fact, there's more than a possibility, is a certainty of a discrepancy between what's promulgated, what's stated, and what the truth is in the population. In other words, in an election poll, what they're saying, certain percent are going to vote for this candidate, that candidate, and what the actual percentage will be on election day. So that's one aspect of this. Now, another term that you see a lot these days are what we call Bayesian methods. Bayesian methods differ from, probably if any of you have taken any kind of statistics, you've learned a statistical approach that statisticians give the fancy name of the frequentist approach. And I won't get into all the specifics of that, but that's just, you know, the tests that you use, and then of course the p-values that are generated, and then what you do with those p-values in terms of interpretation. But there's another school of thought that you should be aware of that's known as the Bayesian approach, which basically takes into account the idea that when we go into an analytical problem or a research problem, there's things that we already know about that problem. If you're doing a study on people who suffer with alcoholism, you're not going into that with a complete absence of knowledge about that particular population. You probably studied it before, you know certain aspects of it. And Bayesians say we should introduce that into the analytical process. The problem with the Bayesian technique, which is why it's not more widely used, is that it is hard to quantify what you know. And frankly, what you know, what I know, what someone else knows can differ one from another. But it is a particular popular method. It's a method that's been more and more popularized, and it finds its home in things like artificial intelligence, about utilizing machines to do jobs that basically learn as they do. do the job. And what they're using is the observations and what they're programmed with, which is what we would call prior knowledge on this basis. And then finally, the research process, which we've already talked about, which is identify the question, the hypothesis, design the study, collect the data, and so on, to eventually come up with results. Now, like I was saying a few minutes ago, the first thing that we want to do is look at our data. Look at the data in order to see, does it look the way you expected it to? Because sometimes, you know, you have an idea about what the study is supposed to show, and wow, the results, there's something, you know, kind of fishy about this, and it causes you, first of all, to not only question your hypotheses, even without analyzing anything, but to question whether the data were collected properly. So we do things like looking at the mean and standard deviation. You're all familiar with these, I'm sure. The range of the data, things that are a little bit more arcane, the skewness and kurtosis, which is basically the shape, measures of the shape of your distribution. And then, of course, we have graphs to help us this way as well. Scatter plots, which are basically diagrams that look at the relationships between two variables. Box plots, which are very, very popular these days, which is a way of presenting the percentiles of your data. And then things called quantile plots, which are a way to look to see whether your data are bell-shaped. And we'll talk a little bit more about that in a minute. So let's talk about dealing with the data if it becomes problematic. And as I was mentioning, we might have outliers or extreme scores that sometimes pop up. Somebody has, you know, if you're looking at, for example, hospital stay for a particular disorder and that you're trying to treat, you might find that a handful of people, or maybe one or two people, stay an inordinate amount of time. That will skew the data. That will skew the mean score. It will also increase the variability. And so we're going to have to, first of all, make sure that we define what we mean by outliers. A lot of people say that if you have data that's outside of 3 standard deviations from the mean, provided the data are bell-shaped, normally distributed is of course the term we use, that maybe we should identify that value. Now, what do we do when we identify a value? What we do is, the first thing we need to do is check to make sure that value was recorded properly. If it was, sometimes people say we should exclude it. And sometimes, as you can see from this last bullet point, people will exclude even things that are relatively closer to the mean than 3 standard deviations. That is absolutely not recommended. My admonition to you is never throw out data unless you can argue that that particular data point or that person or that individual shouldn't have been in your study in the first place because they didn't meet the criteria of the study. For example, you're doing a study on diabetics, and the person that has this outlying score is not a diabetic, but someone with, say, multiple sclerosis. Somehow they ended up in the study. That's a reason to exclude someone, but usually not. So that usually means we're going to have to deal with these extreme values, and there are different techniques for doing that. We're going to talk about those. Okay. Now, when we think about what we're going to do in terms of evaluating the data from a statistical perspective, there are 2 fundamental types of analyses we do. The first one is we have a lot of different variables, a lot of different measurements that we collect, and we want to see if there's a relationship, or better yet, can we predict one thing from another? Can we predict, for example, or determine why a program works on certain patients and not others? And of course, that's going to be a function of usually the characteristics, perhaps their gender, their race, their age, any of a number of other things. And that's really the goal of the study. Why do certain people do well, other people don't do well? And then in other types of situations, we're really comparing 2 interventions. We may have some new approach for reducing, as we were talking about earlier in the TROS study, reducing the transmission of STDs in women who have substance abuse problems. And we have a new approach, a new dynamic, a new paradigm, and we're going to compare it to usually what we do in these kinds of studies, standard of care. So then we're looking for tests for differences between groups or treatments, and we have this pretty standard kinds of approaches. We also organize tests in this regard in terms of whether or not we're willing to assume certain characteristics of our data. And I'm sure most of you are familiar with, again, the normal distribution. Here it is, not that you have, I'm sure, never seen this before. We're assuming in so many of our procedures that we have this kind of shape. That assumption is often fails in the real world. And you might say, okay, well then I guess a lot of the approaches people use are flawed, and in some sense they are. Now there's a couple of things we can do about those. If we have, for example, data that fits not a normal distribution, but it has a skew to it. In other words, there's a lot more people who have people have bigger values rather than smaller values. It's called a right skew. Under those circumstances, we can fix that problem by changing the scale of the measurement. For example, income is a good example of this, or cholesterol level, weight. All of those tend to have right skews to them. How do you fix that? You change the scale. You use the logarithm. And when you do that, very often you get a bell-shaped curve under that scale, and now you can go ahead and do parametric tests. As we know, statistically, they're very powerful. And when I talk about parametric tests, I mean things that you're all familiar with, like t-tests and analysis of variance tests. Those are very powerful. That is, they can detect differences a lot better than if you don't make that assumption about normality. And if you don't make the assumption about normality, then you're using what we call a non-parametric test. So that's the differentiation between these two broad classes of tests. Parametric, where you assume that the data have a normal distribution, or you can transform the data to have a normal distribution, and non-parametric tests where you make no assumptions about the shape of the data. And often people say, well, let's do that. Let's use non-parametric tests all the time. The problem is they're not as good in terms of finding the differences that you're setting out to find. That is, showing the intervention works better than, say, a control. So we don't like to use non-parametric tests unless we have to. The only time I regularly use tests that don't make assumptions like normality is when I have very small sample sizes, because then it's very hard to demonstrate or to assure ourselves that the assumption of normality is met. So again, these tests don't make assumptions about the distribution. We also call them distribution-free tests. And as I say, they're lower, less power, less ability to detect differences, and less flexible than parametric tests in terms of how we evaluate the data. But again, they're very useful for small sample size. So recommendation here, if you're thinking about, do I use a t-test or a non-parametric test, and we'll talk about those in a minute in terms of their names. We always try to use the parametric test if we're satisfied that the assumptions are made. And even some deviations from the assumptions still are okay. The word we use in statistics is robust. A test is said to be robust even if you deviate somewhat from the assumptions, then you're still in good shape. Now, we also organize tests by the type of design we use, or the type of research we're doing. In the type of research that I know many of you are interested in, we're talking about experimental research where we have an intervention. For example, been working on for years now at the VA the idea of smoking cessation. That's a big deal in the VA population because many of the smoking prevalence in veterans is very high relative to the larger population as a whole. So we're doing a lot of smoking cessation programs. So we're comparing those to controls. And then we're recording outcome variables like smoke-free days, for instance, or time to, is we have independent variables, which are the programs or the things that we're manipulating to start the study. And then we have outcome variables or response variables, which we'll call dependent variables, or DVs for short. Now, a lot of you also do survey research, all right, where we're doing, and of course, first of all, before we get to that, experimental research is usually longitudinal. You're basically intervening in one group or another. We often use, some basic techniques like randomization to put patients or subjects into each of the two groups, and then we follow them over time, therefore longitudinal research. But sometimes a lot of times we'll also be doing survey research, which is point-in-time research. We're concerned with that type of research in looking for prevalence of certain things, or relationships between variables, which is what we talked about earlier, where we're looking to see if the independent variable predicts something about the dependent variable. For example, in this study by Kilpatrick, we're looking for the relationship between PTSD, depression, and substance abuse in adolescence. Now, the difference between the two in terms of the type of research design is that in experimental research, we're testing a hypothesis. Does this smoking cessation technique work better than what we're doing now? In survey research, we don't really have hypotheses per se. We're just looking for, as I said, estimates of prevalence or relationships between variables. Although sometimes studies do both, and sometimes in experimental research, we find that, oh, this group, this program works better than standard of care, but it doesn't work well on everyone. Now, why? Which people does it work better on? And then you're into this correlation or association research. So again, just to remind you, we have these two types of variables. The independent variables are the treatments or the interventions, and then the outcomes are dependent variables. And so what we assume in terms of a basic kind of model is that the independent variables either cause or predict the dependent variable. Now, causation is a tough thing, okay? Causation, we have to be very careful. Most of the time, I like to just warn people, don't use the word causation sort of in a routine way. Use the word association, because causation, just because we find a difference between two groups, doesn't necessarily mean that one group caused the effect. It could be that there is differences in terms of the samples and the characteristics of the samples. So it could be that there are what we like to call mediating variables that may in fact define why we found what we found. So, but in other cases, we have an explicit explanation. For example, a drug causing a memory defect. Okay, so let's go on to the next slide. So now let's talk about the ways we pick our approaches, our statistical approaches. And one of the classic ways to define how to do that is by what type of measurement have you made? What have you collected? What is your main outcome variable or variables? And this was defined about 60, 70 years ago by a psychologist by the name of Stevens. And Stevens said, I'll show you all of them at once. Stevens said that we can define different classes of variables according to what we can, how much we can manipulate the data. So we start at the top here with nominal scales, which are basically just categorical things. We can identify or put people into categories, but there's no magnitude, there's no meaning to the even if you assign a number, we can say men are 1, women are 2, or we can say that if you said yes to this question, you're a 1. If you said no, you're a 0. Those are meaningless. Those are arbitrary. So the magnitudes of the values you assign to someone, if you decide to do it numerically, don't mean anything. So categorical data, or sometimes known as, we'll see in a minute, qualitative data. Ordinal scales allow a little bit more manipulation, allow you to do a little bit more mathematically, in that you still have categories potentially, but you now can put them in order. For example, severity of drug dependence from say mild to moderate to severe. Those are meaningful. Even though those are categories, there's an order to them. And what it turns out is that statistically we can make use of that. We can make that is semi-quantitative in a sense, and we can use that. Now, the last two are a lot of people say this is a waste of time separating out the last two, because the last two most people think as purely quantitative, things that you measure like temperature, for instance, or number of packs of cigarettes you smoke per day. But the point is that there is a differentiation based on whether the zero on the scale is arbitrary or not, or put it more simply, can you have negative values? If you have negative values, there's certain things that you really shouldn't do mathematically, like look at ratios. So for example, on the Fahrenheit scale, the Celsius scale, you can have negative temperatures, of course. It could be minus 10 degrees Celsius one day, and maybe 6 months later, it could be 90 degrees. Excuse me, not 90 degrees Celsius, it could be 90 degrees Fahrenheit. So if you look at those two temperatures, minus 10 degrees Fahrenheit versus 90 degrees Fahrenheit, that's a 100-degree difference, but the ratio doesn't mean anything. How many times warmer is it in the summer than in the winter? Meaningless question on those scales. So that's the only reason we make this distinction in terms of interval scale. And so there's a ratio scale to really come to the point where all the data that you collect, you can manipulate any way you want, and that's because there's a true zero. You can't go below zero on a ratio scale. Pretty much everything you can think of, I'm giving you one example, packs of cigarettes per day, but anything you can measure, like the physiological lab measurements like blood pressure or cholesterol levels or white blood cell count, hematocrit, all these different things, pulse, all these different things are on a ratio scale. Now, let's take a look at some of the things that you can do with each types of data. So again, thinking about the two types of hypotheses we were talking about. Nominal data is categorical data. So let's talk about first about looking for the differences between two groups in terms of categorical data. In our smoking cessation study, we might ask at the end of 1 year, is the subject stopped smoking, yes or no? Okay. In other words, and we can define what we mean by stop smoking or cessation of smoking. That's not the point. It's a categorical variable. And now we ask the question, okay, so what do we do with that data? Well, typically most people think about the so-called chi-square test there. But sometimes we have done some things to the design where we match people up, or we have some sort of what we call dependent sample selection, where people are not independently assigned to groups, but they're assigned to groups in pairs. Under those conditions, we have a modified type of design, a test, chi-square test called the McNamara test. Now, what if you're looking for association? Again, you have categorical data, which you have two categorical variables, whether you got the smoking cessation program or not, and the second one, did you stop smoking at 1 year? And you just want to see, are the two, how are they associated? And there's some very crude measures of association, one of which is called the coefficient of contingency. Most people have probably never heard of that because most people don't think about association under these conditions. They merely look to see, is there a significant difference in the proportions in each group in terms of, in our example, smoking cessation. Now, ordinal data, we can be a little bit more flexible because now we have some mathematics, we have some numbers that are orderable, or that's the right word. And so again, we think about, but remember, this type of data can't possibly be bell-shaped, so we have to shy away from using any of the things that you're usually familiar with. And again, we break the type of sample up into the groups, whether it's independent versus dependent. So we might be asking, again, using my smoking cessation example, not whether they stopped smoking, but have we reduced the severity of their smoking? All right, just using you know, kind of a simplistic example, you know, from heavy smoker to moderate smoker to light smoker to non-smoker, if we decided to categorize people like that. So that's an ordinal variable. So if we have, for example, just simply two groups, then we're going to compare the two groups using a non-parametric test. It's usually known as the Mann-Whitney test, sometimes also known as the Wilcoxon test. If you have if you have multiple groups or multiple measurements, then you're going to use what's equivalent to an analysis of variance with a non-parametric version of that. It's called the Kruskal-Wallis test. If you move over to dependent sampling, you can see you have what's called a Wilcoxon test. It's actually technically known as the Wilcoxon rank sum test, sign rank test. And if you have multiple groups, you have what's called the Friedman test, which is an analysis of variance based on the ranks, because that's all you have here are ranks. And then you have a measure of rank correlation that many of you have heard of. It's called the Spearman correlation. Again, you can't do much more than this because all you have are ranks. You do not have real numeric data, and so that's important. Now we go to interval data, and actually, when I call it interval, it's really interval ratio data. And basically, here's where you have all your usual parametric tests. And I'll just, just quickly to run through those. You have the t-test for two groups. You have the ANOVA for multiple groups. And if it's dependent, you have the paired t. And then you can also, turns out, you can do an ANOVA on multiple groups that are matched. Of course, you're all familiar with the Pearson correlation in terms of the measure of association. And then if you have multiple measures, then you have what's called the multiple correlation coefficient. Now, there's another way of thinking about our data, which classifies things according to whether it's quantitative or qualitative, which simplifies that whole Stevens approach. And quantitative variables are very simply those that vary across a range. In other words, you basically have the ability to have values, and the values themselves are proportional to the amount of the quantity being measured. For example, time to relapse after, maybe a person who is on a drug treatment program, and one of the things that you're looking for is, do they relapse, or better yet, how long it might take them to relapse, or the number of drugs they're abusing, for instance. There are two different types of quantitative variables: those that are continuous, for example, like time, versus those that are discrete, which are numbers. And again, the types of analysis are going to vary depending on whether you have a continuous, like time to relapse, versus number, how many cigarettes you smoke. Those are going to dictate different types of statistical tests. On the other hand, we have qualitative data, which are equivalent to what we just called in Stevens' method, Stevens' classification, nominal data. And these, of course, are categories, for example, gender, or whether or not you're a cocaine user. Although we code these things, as I said before, that coding means nothing. And then particularly, most of the time when we think about this type of data, we're usually thinking about what we technically call dichotomous data, or dichotomous qualitative data, which take on two values: male versus female, user versus non-user, for example. And a lot of people like to take continuous variables and reduce them to either discrete or even qualitative. Sometimes people like to say, okay, rather than use age as a factor in my analysis, I'm just going to say, if you're under 65, you're in one group. If you're over 65, and you're another group, because it's much more convenient just to code things like that and not worry about individual age. Many times, if you do that, you're going to risk the possibility that you miss relationships. I'll give you a quick example of a study I did many years ago in patients with malignant melanoma, stage 4 skin cancer, in other words, where we had a choice of either seeing whether a patient would live at least a year under a new type of therapy versus standard of care, or how long they live. It turned out the study went for a year, and unfortunately, none of the patients survived a year. So if we had just simply looked at the qualitative measurement of, did you survive a year or not, we'd have seen no difference, nothing between these two variables. On the other hand, if we, not if, but when we used how long they survive, it turned out there was about a 4 or 5 month difference in survival time. While that doesn't sound like a lot, in a disease where the median survival is about 10 months, that's a big deal. So we were able to see something with a continuous measurement that we couldn't see with with just simply a dichotomous or categorical measurement. So we need to be careful. A lot of people I know that I work with like to do that because it's simple, but be careful. Now, choosing the test from this perspective, we ask some very simple questions, and this is what you should be doing. What type of data do you have? Again, we talked about nominal or frequency data. In other words, how many people in a category. It's the chi-square family. Are we interested in relationships? If the focus is on relationship, then we go to correlation data. If the focus is on difference, we go to family of tests comparing groups or treatments like the t-test or the analysis of variance. And then, of course, then these types of tests are distinguished by how many independent variables we have, because sometimes we have multiple independent variables, and whether the measurements are made repeatedly or on different people. And then, of course, take all of that, and I know this is, there's a lot going on here, and I know in a short period of time, it's a lot to digest, but this is the thought process we follow. This is what it takes to basically learn statistics, is to ask ourselves all these questions. And as I said at the end, we then have the combination or the choice between parametric and non-parametric tests. So, okay, so I'm going to show you that big old diagram rather than whoops, there we go. And so this is that big flow chart for basic statistics, and I'm not going to go through the whole thing, but you can see that we can convert basic statistics to something that's very algorithmic. In other words, you basically ask questions of yourself: frequency versus, in other words, categorical data versus numeric data, differences versus relationships. How many variables are you looking at? How many experimental conditions? Do you have two groups versus more than two groups? Are you measuring people repeatedly, or measuring people at different points in time, but they're different people, and so on and so forth? And then finally, are you using parametric versus non-parametric tests? This is the green diagram I mentioned earlier. I think this is an extremely valuable diagram. I've seen this in a lot of basic statistics books in the back cover. They don't want to put it in the front because they don't want to scare people when they open their textbook. But at the back, after you've kind of gone through the course, now I see where it all comes together. So this is kind of a very useful way to thinking about all the different tests that we have. And unfortunately, I know it's hard to believe, but this is just basic statistics. I say unfortunately, because this is only the beginning, certainly not for today, but it gives you the other issue. Now, I want to finish up a little bit by first of all mentioning about the fact that when we do all these wonderful tests, we can make mistakes. This afternoon, in my lecture this afternoon, when I'm going to talk a little bit about sample size and how to design your studies, we're going to talk about this again. But we have the idea that we have errors whenever we decide to when we do a hypothesis test like this. Type 1 error, the first type of error, the more serious type of error that we can make is when we say that we're comparing, let's say, this intervention with the smoking cessation program with a control. And it turns out the smoking cessation program really doesn't help people give up smoking. But we conclude that it does. That's a bad thing, because it basically tells people, oh yes, this program should work. You should institute it, use it in your place. It has an effect. And unfortunately, you're telling people to do something that they shouldn't do. It's what we would call an error of commission. It's the worst of the two types of errors. And by the way, if you didn't realize this, it depends on your significance level. In other words, how you make the decision as to whether to reject the null hypothesis, which most people set at 5%. And so you'd say, okay, we're saying that we're going to make this error 5% of the time when a treatment or an intervention has no effect. And some people say, well, let's set it a lot smaller. But the problem is, if you set it a lot smaller, thereby not making this error as often, you will make it more difficult to find what you're out to find, that this method or this treatment or this intervention works. So we got to be very careful about setting this too small. So people about 75 years ago came to a conclusion or came to a convention that 5% is about right. Although that's up for discussion recently. Won't get into that today. Type 2 error is the opposite type of error. It's when you say, oh, this smoking cessation program, unfortunately, doesn't show anything. So I guess that's no good. That's falsely accepting or not rejecting the null hypothesis of equality of the two groups. That's not so bad, okay? Well, it's bad, but it's not as bad as type 1 error, where you're telling people to do something that they shouldn't. This is an error of omission. And it obviously misses a significant effect of a treatment, which makes you go home and start crying in your pillow and saying, okay, I guess I better go in another line of work. Well, not so fast, because a lot of times people say, well, maybe I need to just do another study or a bigger study. And that's usually the point. We missed a result because we didn't have a large enough sample size. That's what we're going to talk about this afternoon. Now, the last thing in terms of statistical tests is the bigger picture. Everything we've talked about so far is what I like to call a univariate test. Essentially, you've got an intervention and an outcome, as opposed to a multivariate test when you have multiple dependent variables or potentially even multiple independent variables. For example, study by Hay et al. Looking at what causes or what predicts adolescent multiple drug abuse. And he looked at 19 different predictors using what we call a multiple linear regression model. So regression models, multiple regression models are what we employ when you have multiple predictors, multiple independent variables for looking at some particular outcome. Kipke in 1997 looked at how long someone stays homeless as a function of drug and alcohol abuse as potential predictors. And then again, we also have the possibility of combining dependent variables about, smoking, drug abuse, alcoholism as a group, and what predicts all three of those in a single model. That's what we call multivariate multiple regression. That gets pretty complicated in terms of the math and the actual manipulation of the data, but actually has the same concept. Are certain independent variables predictive of, again, now multiple dependent variables? Now, lastly, let's not forget missing data. And one of the things that people often ask is, what about missing data? What do we do with it? Well, it really depends on what the missingness, which is a word I think only statisticians could love, make the characteristic of the missingness. We have the simplest type of missingness, which is completely at random. For example, if you had multiple versions of a questionnaire and some people got one version and other people got another version, well, obviously, not everybody fills out every version of the questionnaire, but they basically all give you the same results. Do we care about that? Probably not at all. Or if you're doing a laboratory experiment and somebody drops the test tube and that particular sample for that patient is lost, is that going to mean anything? No. Missing completely at random means it's ignorable randomness. And if you can argue that, then you basically just ignore that data and move on. But unfortunately, that doesn't happen very often. Usually when something is missing, it depends on some characteristic. So suppose we had students at a school and we were asking them some questions, we were evaluating them over time, and some of them graduate. Well, obviously they're unable to respond once they've graduated because they're no longer at the school, and whatever we're looking at in terms of perhaps maybe smoking, whether they smoke or use marijuana or something like that. So that's missing, but it's somewhat unrelated to the outcome, because it's not like certain people refuse to answer because of the outcome. but they're just missing because of some other characteristic, namely they graduated. So often in those cases, we're able to handle those fairly simply. The hardest type of missing data is what we call non-ignorable non-response. It's where the missingness depends on their characteristics which relate to the outcome. For example, in a survey of people's income in a public health school, this is an actual study. The people who didn't respond are the people who make the least amount of money. For whatever reason, you can argue why that happened. So the missingness is related to what you're trying to determine. So all you're getting is the response from the people who make the most money. And clearly that's going to affect the outcome. So that's going to create a bias, and we have to deal with that. Now, obviously, we're not going to have the time today to talk about it, but just wanted to give you the ideas that this is something not to overlook. And then lastly, just going back to the multivariate, I guess that last slide probably should have been at the end. You know, one of the things we like to do is look at the data in various ways when you have multiple. If you haven't seen these things, these are kind of neat, particularly that one on the right where you have these contour plots and you can see data. Obviously, 3 dimensions is as good as it gets. But unfortunately, beyond that, our brains kind of get fried when you try to think about 4, 5, and beyond dimensional data. And the type of things that we look at in multivariate data, besides the things we often measure, are things like latent variables, things that you can't directly measure, like a psychological construct, personality variables such as rebelliousness, depression, and risk-taking, or attitude. You can't measure those. So what you do is you measure a bunch of other things, characteristics, and then you use techniques like factor analysis or principal component analysis to come up with a construct that you then could call rebelliousness, depression, and risk-taking. We did a study a while back at Drew in measuring a concept that the investigators wanted to call religiosity. In other words, how religious someone was. And this was being used to determine in a study why gay African-American men were practicing safe sex or not. And the thought was their religiousness or religiosity might play a role. And how do you measure religiosity? How do you define that concept? And we measured a whole bunch of aspects of their personality and their belief in God and things like that, and put it into a variable using this technique, which is part of the process of understanding why some practiced safe sex and others didn't. And then, of course, we have other types of variables that come into play in a lot of these analyses, covariates, things that can influence the outcome. We'll just skip through these. And one last thing here, I forgot to show you the example. Poor versus good responders to treatment with education as a covariate, heavy versus light smokers with personality as a covariate. These are known as mediators again. These are mediating whatever relationship you have between, let's say, a program that you're undertaking versus the outcome. And so you always want to consider these extraneous variables that can influence outcome, but are not under your control. And so this is a really important aspect of all kinds of sociological type research. And then finally, we put this all into a construct which looks really simple. It's called a general linear model. This actually covers everything I've just spoken about for the last 45 odd minutes into one big model that looks really simple. And you know, mathematically, I guess you could say it is. You have a dependent variable, and then you have everything else, which is on the right side of this thing. And it's a conceptual way of organizing everything in terms of independent variables versus dependent variables, the level of measurements, whether you have a single variable like, you know, single outcome or multiple outcomes, and whether you have latent variables and so on and so forth. All of this can be written in one big, I guess, the unified field unified theory of statistics, you're looking at it. Everything could be thought in these terms. And there are programs that can analyze any data this way. And obviously they're a little bit hard sometimes to interpret, but we can think of all statistics in terms of this idea that the world is linear, the world is normal in a statistical sense. In other words, the relationships between X and Y, as you can say, is linear, okay, whatever X and Y represent. And the what's left over, this E, the error, if you will, is normally distributed in the very real sense that we talked about. That's how statisticians like to think about the world, as being linear and normal. That's an approximation, obviously, and sometimes we're right and sometimes we're not. And so here's the bigger picture again. In a broader sense, this is where everything's in it, including the multivariate techniques. I'll just leave it to you to digest this at a later time. And we have some references here that you can look at in terms of the different ideas in terms of basic statistics versus multivariate statistics. There's also a book I have on my shelf that's called Textbook of Parametric and Non-Parametric Statistical Procedures. It's about that big, and it basically gives you every one of these procedures and tells you how to use them, how to do them. If you want a book on your shelf that has all of that, it's by a guy named Shulkin, and it's pretty thick, pretty fat, probably something that might also make a good doorstop as well. All right, and then of course we have some software. All right, so that is that. Okay, so questions? A lot in 45 minutes.
Okay, Dr. Lee, I enjoyed your jokes, and that's what I don't like about Zoom is I feel like it's so hard because you're making jokes and you're like, are they laughing? But I was laughing at the first one and the last ones. But yeah.
I encourage my students to send me chat, LOLs in their chat box.
Yeah. Yes, that goes for anybody here too. Feel free to send an LOL.
Great presentation, Martin. Do we have time? We have some time for questions. Does anybody either want to unmute or ask a question in the chat?
Thank you so much, Dr. Lee. We ran a little bit behind today, but hopefully we'll in the afternoon.
Okay. All right. So I'll see you guys all later. I've got to run to another class. So thanks a lot, everyone.
Thank you so much. Thank you. Thank you. So next we have a break until 11. So feel free to take care of yourself and do what you need to do on our break. And then we'll resume at 11, at which point we're going to have Dr. Sherry Larkins give a talk on survey design. So I will go ahead and share my screen just to remind everybody to resume at 11, and we hope to see you again in a little while. So thank you, everybody. Dr. Larkins, I see that you're on, so I'm going to make you a co-host, and then you can share your slides. You can still share your slides even if you're not a co-host, but at least this way you have full privileges if you need anything else. And I'm not hearing you, and you're not muted, so there might be something with your volume. Still don't hear you. I know. Okay.
Sometimes you have to unmute through, it depends on how you're unmuting. Sometimes you can do it through your computer versus the Zoom. I would try both.
Okay. I think she logged off. She might be.
Did she? She might be logging in. Sometimes people have, when, you know, you use the icon down at the bottom, but then you can also unmute yourself. Well, hopefully she'll be back in.
Yeah.
You've been doing a great job, Christina.
Oh, thank you. Thanks.
Yeah,
I set it
really
on auto
good.
record. And so I was thinking that it was auto recording and then realized that it wasn't. So
Oh,
whoops.
well, you've got a lot. It's hard to have all the pieces, you know. I mean, there's so
Oh my
much going
goodness.
on, but yeah, you've done a great job
Oh,
moderating,
thank you.
keeping it flowing.
Thank you so much. Thank
It's
you.
exhausting.
What's that?
I know it gets tiring.
Oh,
You deserve a
yeah.
break. Maybe you should run and take a break.
Oh, thank you. Thank you. Yeah, no, I've been stretching and I got some coffee. So yeah, thank you.
Coffee is the most important.
Mhm.
See if she's back on. Oh, here she comes. So let's hope she can get
Sure
on.
that yeah, I want to make sure she's set up as well.
Can you hear me now?
Oh, yay.
All right. I didn't use the link. I just hand entered in the meeting ID and I don't know.
You
I'm in the
know,
office. So who knows what's happening?
It's also funky.
Yeah, man. Okay. Well, sorry that the link didn't work.
That's okay. That's okay. It happens occasionally.
Yeah. Okay. Well, I was trying to stop the recording for the break as well, but for some reason it's not letting me. So I guess we'll record the break. I guess.
Oh, I wouldn't worry about it.
Yeah. All right. We're just gonna let it go. So Dr. Larkins, are you okay with sharing your screen and your slides?
Yeah, let me just
Okay. Let me make you a co-host again.
Perfect.
Yeah. Yes. Okay.
Okay. Am I up?
Yep. Fabulous.
But you probably want to hit, and people forget to do this, but you want to hit the view instead of the slide sorter.
Good. Okay. Am I?
Slides.
Okay. So let me see.
Start your slideshow. Yeah, you're up, but you just want to start your slideshow so that we get the full slide instead of the sorted, the sorted image.
Oh,
you know what I mean?
Okay. Oh, where do I do this? Let me see. More
just
slideshow.
Go to the slideshow bar at the top.
So I think you might presenter view now. But I think if you go to end slideshow, and then on the top tab, see home, insert, design, layouts, click on slideshow.
Yes, keep going.
Okay. Design.
Keep going.
Oh, transition. Oh, slideshow.
Keep going.
Okay.
Keep going.
And then from the beginning.
Start from current slide. Yeah.
Or yeah, whatever. And then do you have notes that you need to see in your slides?
No, I'm okay.
Okay.
So is it showing properly?
Hit from beginning.
Is at the beginning. What?
On the lower row, go to the far left.
Okay. Right here, yeah. Do you see survey research considerations up?
Yeah,
yes,
so you
yes. But what we want you to do is get the full slide view. So just go up to the second row and you'll see the first icon says from the beginning.
Right.
The row underneath the brown bar.
Right. Oh, wait,
from
okay.
Beginning.
Mine just goes to return to the previous slide. Is this right?
Oh, that's weird. Yeah, and for some reason, we're not seeing anything happening on your screen too.
Okay, so if I go like this, are you seeing my slides move?
No, for some reason it's not.
You have to, so Sherry, you don't see under the brown, on your screen, do you have the brown bar on the top?
The other choice, Sherry, is where it says, see at the bottom right, it says 65%. Right to the left of that.
I'm not seeing that. Let me just get out. Okay, yes, sorry. I was, okay, so yes.
Right to the left is, keep on going to the left.
Make it a full
right there,
slide.
got it. Right there, got it.
I
click that one. There you go.
Oh, that one's presenter view though.
Okay.
So then I think maybe if you
look at display settings.
Swap presenter view and slideshow. Let me try that.
Try that.
Okay.
There we go.
Oh, there
you go.
Yay!
Yay!
Mission accomplished.
6 minutes to spare.
Yay, awesome.
We don't
Cool.
actually start the slideshow, so what we get is a reduced size of the slide, and it's often on a laptop really hard to see. So you want to start the slide session. You did it. Perfect.
Yay.
Hey,
Great.
phew.
Fantastic. Woo.
You always need a couple
Zoom is-
minutes with these things.
I know, and Zoom is just so awful.
Horrible.
It's like, how many times do we start talking and we're muted?
I
Yeah.
know.
Yeah.
All the time.
I know. I was laughing at his jokes. I felt so bad at Dr. Elise, but I didn't want to mute myself and be the only one laughing. So, but man, it's rough. It's really rough. But we will get through it.
Hopefully
Do
there'll
you
be an
all
end
think-
in sight.
What's that?
That hopefully there'll be an end in sight.
Yeah.
Someday.
I'm teaching in spring and I'm not looking forward to, I want it to be back in class
I
starting in
know.
April, but
It might
otherwise,
be.
oh,
So far, they've just issued the shutdown through March.
right. I hope I eke in at least. I think it's hard to have 150 kids a few days a week on
Oh my
Zoom.
God.
Oh, that's a big class.
Oh,
no.
that would be horrible on Zoom.
Oh yeah.
I know. I'm trying. I'm not going to worry about it till it gets closer, but I'm going to have to figure out how to make it more engaging.
Definitely.
You know.
Definitely.
You know.
Don't worry. You can do this thing that a lot of people are doing where they tape the lectures. They have the students listen to the lectures and then just the other lecture, if you have 2 a week, whatever, is interactive, where they have to participate based on the prior lecture.
Right. Good idea. I mean, it's, you know, all these things I've been watching on like how to make great engaging breakout rooms and stuff, but I'm like 150 kids. I'm more of like a technical, my job becomes technical of trying to get them in and out. It's like, it's not content, it's like process, it's people management. And I'm like, you know, it takes away from actually, so, you know, figure this out.
Yeah, with 150, it's more like a webinar. Like you're not really doing it. Wow. That's hard. Huh. Yeah. Well, do you all have any suggestions on maybe to keep us on track? Should I maybe give like a 5-minute warning to speakers? Do you think that might be a good idea? To send them either a private message or do it like verbally? Do you think? Should I interrupt their talk?
You're okay to interrupt me. And plus I don't have that many slides, so I'll have a little break for these guys before Steve comes on.
Oh, thank you. Just in general, I think one person emailed me too, and they're like, oh, we're really behind. And I was like, oh yeah, we are. So just to kind of help folks get back on track. What do you think?
Absolutely. Yes,
you think verbally?
Definitely. Try the chat, or you can try chat, or you can just say, I'll be sending you a 5-minute, you know, reminder in chat, or, you know,
that's a good
idea. Tell them so they'll know to look.
Okay, that's a good idea. Thank you for coming on. We'll send you a 5-minute reminder. We'll send you a 5-minute reminder in chat, Dr. Larkin.
Okay. I'll be good. You don't have to worry.
You'll be my first guinea pig. Yeah, no, I'm not. And I think we've been pretty good. I think we got back on track. But
absolutely.
Yeah.
Yeah, definitely.
Get there, working out the kinks for the first one. So yeah. Okay. Great.
So Christina, you can just tell me, you know, if you guys are giving people a 5-minute leeway, or if you're starting right on time, just give me the green light and I'll be ready to go whenever.
Okay, sounds good. So it's 10:59, so we can give folks another minute. But yeah, I feel like we probably should start on time to kind of keep everybody. Yeah, just watching the clock. Let's see if most people are back. I know a few folks logged off and they said they'd be logging back on. So yeah, but most folks stayed on. So I think that it would be okay if we start. Dr. Friedman, what do you think?
Okay, I'm ready. Yeah.
Okay.
Great.
Okay.
Great. Well, thank you,
Sherry,
Christina.
Sherry,
Yeah.
I'm going to give you a little introduction. I know we're anxious to get
Great.
started, but I want to praise our speakers. So Dr. Sherry Larkins worked for Friends Institute and UCLA Integrated Substance Abuse Program since 1998. She currently directs the ISEP's international training and educational efforts, including 2 SAMHSA-funded projects in Southeast Asia. She also leads the training and education efforts of ISEP's visiting scholars. Prior to her current position, she directed training and technical assistance initiates for the Los Angeles County Department of Mental Health. She's led multiple epidemiological and clinical research studies, including one investigating the spread of HIV and other sexually transmitted infections among substance abuse users, and others that focus on treatment interventions. Dr. Larkins has been involved in substance abuse research for over 20 years and completed her doctorate in medical sociology at Rutgers University in 1999. Her research interests include stimulant abuse, substance abuse treatment for marginalized populations, sexual risk behaviors associated with substance abuse, and qualitative methodologies. Thank you very much, Dr. Larkins, for joining us today, and we look forward to hearing your talk.
Great, thank you, Dr. Friedman, and thank you, Christina and Chris. I appreciate you inviting me, and welcome to the class. So I know that this series is on sort of methodological skill development, and one thing I'm going to be talking about today and focusing on is really survey research. And I would say this is a fairly sort of basic introduction of what survey research is, how it fits into the larger kind of domain of research more generally. And then we'll talk a little bit about, whether you're designing your own survey research or whether you're using existing, more like whether you're doing primary data collection and developing your own, or whether or not you're using ones that are already validated and tools that are used in the substance abuse field or in, HIV or sexual risk or mental health field. There's a lot of things, survey work that's already been developed and validated. And so we'll sort of talk about that process today. So when you think about how surveys fit in and really sort of data collection fits in, I sometimes just like to use what we call sort of this wheel of the scientific process. And this is really more on what we would call deductive research, where you start with, up at the top with, what's the question you have? What are you interested in? What's driving your area of research? And within substance use, you may have specific interest in special populations, like, what are things that specifically affect women, or what types of interventions specifically are targeting women's issues? Or maybe you're interested in other, maybe specific drugs of choice. How do opiate users look different from stimulant users? And so in the process, we often develop a question, a research question, and then we sort of, refine those into some pretty clear hypotheses. And this is where we select which kind of variables we're going to actually be looking at. And we start to develop an instrument that addresses that specific research question. So our instruments are tied to the questions that we have. And in this process, we also think very carefully about how we actually sample the population. Because ultimately, what surveys help us do is they allow us to take a smaller group and extract or extrapolate to sort of what we think is happening on a bigger scale. And so the area in red at the bottom you see is really when we collect sort of systematic observations. And in some cases, that's a survey. Surveys are the most common or popularly used tool or instrument to investigate in social research. But there are, there's experimental designs, there's other types of data collection, but surveys are the most common. And from our surveys then, once we collect data, we kind of go around the circle and you can see, how we analyze data, how we, take observations and how we measure, and then how we analyze or look at that, and then how we interpret those results, and ultimately how we disseminate the findings. And this is kind of like the process of science here and generating science. And so you can kind of see where surveys fit in. And so something that's important to think about in this process, I started with sort of the developing hypotheses and surveys, but something that we always do with our surveys that we develop ourselves is we pilot those instruments as well. And so, and then again, who's the target population you're trying to find something about, how are we collecting those data and analyzing it, and ultimately, how do we present and publish findings? So you can see at the bottom, I think this is me when I get called for surveys. And right now, in political season, I can't tell you how many calls we all get on this topic, and either asking for money or asking for our opinions. So surveys are, as I said, the most widely used data gathering technique. And the idea is that we're from a smaller group or a sample, we're able to extract information by asking a series of questions through what we would consider a more deductive process. There is also sort of inductive research, which lends itself to more exploratory and qualitative work. And I think you're going to be talking about some of that as well. And usually when we think about surveys, the real goal in sort of ideal, more scientific way would be that we would have a random sample of people, we would have valid and reliable instruments, and then we would have, a sort of sophisticated statistical analyses of the data. And this is really the most efficient way to describe large populations, to track trends, to find out what people's self-reported beliefs, their opinions, their attitudes, even behaviors. And one of the strengths, in some ways, when we think about which methodologies really are the methods that we use are driven by the research questions that we have and what we already know and what we are looking to find out. And surveys are very good because they can tell us a lot. They may not go as deep and detailed as some other types of methods that are more qualitative in nature or more open-ended, but within one survey, we can find out a lot of information. We might be testing on multiple variables. There might be multiple hypotheses that we are able to capture through sort of
early. Yeah, it's a substance abuse conference.
Okay, so mostly questions on surveys are closed-ended and not open-ended. Usually the idea is that we're able to kind of quantify something through a closed-ended process. And surveys are really good about answering these what, where, and when questions. Sometimes the questions that require a little bit more of the why or how are not always best answered through a survey. Because surveys typically have a fixed choice response or are closed-ended, they don't really allow us to sort of get into sort of some of the deeper things that are happening. So again, what, where, and when are good things we can capture in a survey. And when we want to know sort of why something happens or how something happens or stuff that's that involves, I think, more sort of depth or details, we often have to complement our survey research with more qualitative research. And when we are going into the field and doing survey research, we kind of distinguish between primary data collection and secondary data sources, or primary data and secondary. And sometimes, we use existing data. Maybe the research questions that we have, we can best get at through existing surveys, ones that have been validated and they're reliable, and we've seen them used very effectively in other examples. And so there's no reason to sort of reinvent the wheel. And in other cases, the work that we're doing is distinct and unique enough that existing surveys don't exactly capture what we want. So we have to sort of develop our own. And one real benefit of surveys is that they're often strong on reliability and measurement validity. So we can get some uniformity and standardization, which doesn't really happen in more kind of open-ended or qualitative work where it's kind of hard to put things into boxes. By design, surveys we're putting things into boxes, and that allows us to be sort of more uniform in some ways. And there's a lot that we can cover in surveys. We often talk about, particularly in substance use work, we talk about behaviors. We ask, you know, how often do you use condoms? You know, frequently, you know, often, sometimes, rarely, never. Sometimes we are asking them about how frequently they use certain substances, maybe how frequently they inject. We're asking questions about their specific behavior. Have you ever, yes or no? But we also can find out a lot about people's beliefs or their attitudes. Do you approve of this? On a scale of 1 to 10, how much do you enjoy drinking alcohol? What's your belief or opinion about this? And sometimes our surveys are trying to capture more something that's descriptive, that we really want to find out who it is, how old are you, how frequently, you know, what's your age, what's your basic demographics, what social class do you belong to? And finally, surveys are really good at capturing what people know. So sometimes we want to know, what's your level of knowledge on something? You know, we want to find out what's people's understanding of something. And in this case, you know, how is HIV transmitted? We want to know, do certain populations differ in their knowledge or their understanding of certain things? So these are kind of the common behavior, attitudes, beliefs, opinions, and knowledge that we can capture in a survey that's harder to capture in other types of methodologies. So as you go into the field, or as you're starting to consider the types of survey research that you want to do, there's lots of things that you need to consider in this process. And some of it is, you know, your research objectives, like I said, who's your target population? But we also need to think about, is this a cross-sectional or is this a longitudinal design? Are we going to go into the field one time and collect data because that meets our research needs? Or are we going to need to sort of repeat measures? And if so, at what sort of frequency? And is it going to be the same types of measure at every single point? Sometimes we need to know how are we going to sample? Sometimes we do a power analysis. We try to understand ahead of time, how many people do I actually need to distribute or disseminate this survey? How many people do I need to conduct this research on in order to say what I want to say? And that tells us how big or small a sample can be. And as I mentioned before, can we use a previously used data collection instrument, or do we need to construct one of our own? What questionnaire, what types of questions will we be asking? Is it going to be open-ended or closed-ended? And also, how are we going to do this? Are we going to do something in person? So we're collecting survey research, but we're doing it sort of face-to-face with a research assistant and a research subject. Are we doing something over the phone? Are we doing something in mail or even over online? And who's going to collect it and how are they going to be trained? We need to make sure that people are trained to collect data in a way that is consistent and is uniform across the survey. And so usually before a research assistant or somebody sort of enters the field and starts collecting data, we want to ensure that they've been trained on how to do it appropriately. And then again, what's the time frame? So, and when we talk about the content, what do we want to ask and how do we want to ask it? So I mentioned behaviors, experiences, attitudes, knowledge, beliefs. And we also need to think about, as this table shows, are we talking about something that's in the past, current, or in the future? So even though we hit these things like, you know, behaviors, and we want to know, you know, do you currently use condoms? Is it current, or are we saying, you know, previously? In this case, when you were in elementary school, did you ever hit another child during a fight? So in some cases, our surveys are designed to really be like retrospective or looking back. In some cases, they're designed to be looking forward. In some cases, they're designed to tell us exactly where the person is at the moment. Your research question will kind of drive that past, present, or future sort of tense of how you are designing your survey. And I'm going to walk through a few examples right now of some surveys. These are ones that are sort of standardized surveys, and you can see that they all look a little different, and even the layout. Sometimes if a survey is going to be self-report, where it's self-administered and someone just hands you a survey and you do it yourself, sometimes the layout is designed in a way that's very easy, so there are no questions. Sometimes if it's an interviewer administered, where a research assistant or a researcher is administering it face-to-face, sometimes it's laid out a little bit differently because the patient or the client isn't, or the respondent isn't the one answering on paper, but they're just verbally giving the information. So this is an ethnic acculturation scale. And the goal of this overall scale is really to find out sort of how ethnically intact someone is. This one is targeting people who are of like Latinx populations. It says, do you mostly speak Spanish or mostly English with the people on the list? So they want to find out, you know, what's your most common language, either at home, with your spouse, with your children, with your family members, with other relatives, friends, neighbor, or people at work. And as you go through this list, you can see from top to bottom, it's the most intimate person typically in your life, all the way to kind of the most distant, which is people you work with, and then people you live close by, people who you are very close with, your parents, family members, and so on. And then asking questions about how, you know, do you write in English or Spanish? How often do you listen to radio stations in your certain language? And I think, can someone mute their line? Thank you.
Oh, it's bad.
Thank you.
Okay,
so in some cases, we see instruments. Here's another example of a survey. This is actually a scale that's like within a larger survey. And this is really just about someone's, how they feel they are in terms of how outgoing. This is whether they're an introvert or an extrovert. And you'll see this is an example of questions. I see myself as someone who is talkative, finds fault with others, doesn't, you know, is reserved. And you write on a scale of 1 to 6, you circle which you think you are. So here's an example of an existing scale within a survey. And when we talk about, in a minute, we'll talk a little bit about what a scale is and how it's, you know, it's part of this survey research. And a scale gives you sort of an average. You'll see all of these numbers are marked. And at the very end, we can say this person on average ranks themselves as a 1.3 or a 1.7 on average, which tells us that they see themselves as a little bit more outgoing. This is one from the DAST, which is a drug abuse questionnaire. And this one is you can see now, the ordering is a little bit different. It's only yes or no. It's not sort of a scale. It's not a gradation of a response, but it's just yes or no. Have you used drugs for other than medical reasons? Yes, no. Do you use more than one drug at a time? Yes, no. And you go through, and the number of yes responses tells us something, okay? And because this is a validated instrument that's been used a lot, it has psychometric properties, and we know of other research that shows, you know, when somebody has a 3 or more, what that means. How closely is it tied to like abuse or dependence criteria, or a severe, a mild, moderate, or severe substance use disorder. So sometimes these instruments can tell us, it can be sort of tied in with other diagnostic criteria in the case of the DAST. And this is another sort of example of something called the ACEs, and this is an index. Again, something that's used in survey research that asks, really asks people, this is really about exposure during childhood, the first 18 years of your life, exposure to traumatic events. And it goes through a list of, you know, as you were growing up, did you live with someone who was depressed or mentally ill? Did you live with anyone who was a problem drinker or alcoholic? And you go down and you see that there's pretty significant trauma. Before 18, did a parent or adult ever hit, beat, kick, physically hurt you in any way? So asks about a range of sexual, sort of sexual trauma, potential sexual trauma, as well as physical abuse, emotional abuse. And because this is an index, this one actually does something a little different. It gives us a single score at the end. And we can see that this score is actually tied with a lot of future outcomes and people's long-term risks for engaging in certain substance use behaviors or sexual risk-taking, all really tied to this kind of early trauma exposure. And this is something that's called an index, and again, a validated instrument that is already in the field. And here's one that's really, really internalized homonegativity inventory, looking at, again, this is this Likert scale, but is sort of a validated instrument that really is trying to get at how proud or how much shame does somebody feel about being gay and identifying as gay, and how does that feel for them, and looking at this tied with other types of behaviors and trying to see if there's some predictive value in whether or not someone feels sort of good about themselves or okay about themselves, or whether they feel a lot of sort of guilt or shame about their sexual identity and behavior. And so again, another sort of validated tool or instrument that's already used in the field. And here's just a final one, is something that is important to sort of point out here. This is the CRAFT, which is a screening tool for youth or adolescents for substance use. And we see a part A or part B in this sort of questionnaire, which is part of a survey design. Is part A is really the pre-screening questions, what we call contingency questions. When you're designing a survey, maybe it's not, the survey isn't relevant for every person, or only some parts of it are. So we have these, what we call contingency or pre-screen questions. Did you engage in this behavior in the past 12 months? If the answer is no, skip to a later question. And if the answer is yes, a new series of questions opens up. So we sometimes, again, call these contingency or pre-screen questions. So these are just kind of some things to keep in mind when you're talking about constructing items. Is, you know, we first want to find out, is there stuff in research already that we can use? Are there validated items? Is there previous research? Do I need to don't reinvent the wheel. So do I need to start with something fresh and new, or can I use something that exists? If we are in the process of developing our own survey instrument, which is always a fun experience and everyone should do it at least once, we want to keep in, very clear and sort of simple statements. And we always have to consider kind of people's cultural background and their literacy levels. So typically we have surveys that are no higher than 8th grade reading level. That tends to be the reading level where most people are comfortable. So in some cases, depending on your target for the survey, we'll want to actually have a reading level that's even less than that, maybe between 5th grade and 8th grade. We want to consider those skip patterns of those pre-screen questions. Skip patterns can be really confusing for people. You've probably filled some out yourself and you're like, oh, where do I go? Does this make sense? Do I need to do this part? So layout is really important, that it is easy and clean, and that there's no ambiguity. And when someone marks something, that we can say with certainty that that is what they meant to mark. Short items are good, and avoiding jargon or slang, words that really kind of can have different meanings in different parts of the United States or even in different neighborhoods. And sometimes we try to be very general, and in surveys, we often want to be very specific because we have to ensure there's no ambiguity. So in this case, do you exercise regularly? Well, I might answer that, yes, I exercise regularly. And for regularly, that means for me, a couple times a month. And for someone else, regularly means at least a few times a week. And so we often need to have clarity when we design these. Emotional language, avoiding emotional language, and using neutral terms. One of my many jobs to get myself through graduate school was I used to write test questions for the like sociology GRE or the SATs. And I always used to get like feedback from the reviewers. They used to pay me $6 a test question about 25 years ago. So I don't know what they pay now, but anyway, they used to, I used to get feedback, and my feedback was always, you know, you need to tone down certain words. So I would be writing a question about, you know, a political sociology question. And it was, I would say, in 1982, during the conservative Reagan administration, this immigration policy, blah, blah, blah. And I would get the feedback, you can't call something a conservative administration. And I'd be like, but it was. And they would say, no, you have to tone down your language. Your bias is showing. So you often thinking about how people frame this, the political ads are the best because they violate all of these standards. Sometimes you'll see things that will say, is, you know, is Gavin Newsom's terrible plan for reopening the economy one that you're in agreement with or not? And so by using certain language, you know what the motive is and who's interested in slanting a particular opinion. But our goal in science is not to do that. Our goal in science is to be as neutral as possible. We also want to avoid prestige bias. So you'll sometimes see especially with risk behaviors, or with whether it's smoking behavior, or whether it's, you know, substance use or alcohol use, you know, all sorts of behaviors, like whether you wear a seatbelt or a bike helmet. You know, some surveys will start out and say, you know, scientists universally agree that climate change is real. Do you believe that the air quality X, Y, and Z? And so when you start out with a sort of a prestige statement at the beginning, doctors have shown that using seatbelts is an effective way to reduce death by car accident. How often do you use a seatbelt? We notice that when we have a prestige bias or STEM question at the beginning, that people are more often likely to say, they're more likely to endorse it. They want to look like, oh yeah, that's right, I do that. But when we don't include those, we actually see people respond differently. So we have to be careful in how much prestige bias. Something called double-barrel questions, where you actually are secretly sneaking in two questions. You know, do you always pay your federal and state taxes? Well, some people pay their federal and not their state, or they pay their state and not their federal. So the question only gives you one response choice. We used to have an instrument on, it was a sexual risk-taking one, and it was around anal sex. And the instrument was coded where they included within anal sex, they included fingering. And people, this was, you know, we would have men would come into the clinic. We were looking at HIV risk behaviors. We were looking at substance use behaviors. And they would say, you guys are throwing in a whole bunch of stuff there. Like, I might do some of these sexual activities, but not all of these sexual activities. And you're lumping them all together as if they're kind of the same thing, and they're really not. And what we realized is we weren't very discerning in the questions that we were asking as we were trying to get risk. And there are differences in each of those behaviors with risk. So we spread them out into additional questions. You want to avoid double negatives. I'm going to show you a couple examples in a minute where you use words like prohibitive, and it becomes unclear. Do you agree or disagree with this? And sometimes, again, am I disagreeing with something that's negative, which means I actually agree? And again, one thing we try to avoid in some cases is what we call a response set. So when you go into your doctor, I don't know about you guys, but my doctor says, do you have any of the following? They list all of these things and it's yes or no. I just circle no all the way down, and that's called a response set. it means that people aren't looking at each of the individual questions. They stopped looking because you kind of ordered them in a way where they just say no to everything. So we kind of want to avoid that. We want to often switch up the questions so that people actually have to read the question in order to answer it. So I'm hoping a few of you unmute your mic here. We're gonna do a little mini test quiz now on the questions that you just saw. So, do you disagree with those who believe in the death penalty? Do you disagree with those who believe in the death penalty? Someone unmute and tell me what's confusing about this. Do you disagree with those who believe in the death penalty?
Double negative.
Yeah, it's confusing, right? It's like, do you disagree with those who believe in it? So does that mean I agree? I'm already confused. Thank you. Do you eat fast food often? Do you eat fast food often?
It's too general. What is often?
It's too general. Thank you, Tanya. What's too general about it? Give me the word that makes it too general.
Often, like, does that mean 1 to 2 times a day? Does that mean?
Right.
Yeah.
Right. Does it mean 2 times a day? Exactly. Or does it mean with, for someone who's healthier than I am, they probably think, eating it once a month is often. But for me, 2 times a day is often. Do you support providing health benefits to those who are unemployed and poor? Do you support providing health benefits to those who are unemployed and poor? What's the issue here? It's confusing.
Unemployed and poor, be 2 different populations.
Yeah, we're including 2. You might actually really think that we should give health benefits to people who are unemployed, who lost a job. But you might think, oh, well, people who are poor, I'm not sure. Maybe, are they working poor? Are they homeless? What do you think? So again, you're right. This is a double-barreled. We're asking too many things here. In the past, how many times did you party with your friends? In the past month, how many times did you party with your friends? What's confusing about this one?
Party could mean different things to different people.
Yeah, what does party mean? Does that mean I use blow with my friends, or I had a margarita with my friends? There's a range of what this means. How do you find our hotel service? Excellent, good, or fair? What am I missing here? What if I had super crappy service? Is that on there?
The scale isn't complete.
Yeah, it's really not complete. It's actually forcing me into sort of a biased position so that everything is either okay or good, but I'm not allowed to say anything bad. Did you do your patriotic duty and vote in the last election? What's happening in this question?
It's judging.
Yeah. So by the way I asked this question, did you do your patriotic duty and vote in the last election? Do you think I'm going to get people who are going to inflate their voting or deflate their voting?
Definitely inflate.
Yeah, people are like, wow, it's patriotic to vote. So people are going to be more likely to say. So again, we're kind of like using some leading terms here, and it's going to produce sort of a sort of biased outcome here. So thinking about, again, the questions that we ask and how we do layout is important. We talk about, those skip or contingency questions, but also a typical survey has an order to it. Like we talk about opening questions, middle questions, and ending questions. And we often want to move from the general down to the specific. And we have to think of very threatening questions be coming later, and particularly in the work that we do, where we're talking a lot about behaviors that are not legal, and we're especially in communities where people have really been criminalized for substance use, and it hasn't been seen in a public health context, but in a justice context. There's a lot of reasons why people aren't going to want to be honest about their behavior. And if the first questions we ask are, do you deal drugs to support your substance use behavior or something? If we start getting into things like this, no one is going to answer yes to these questions. But if we start with very general, broad questions and we can increase the sensitivity, we find that we get more authentic answers. You know, asking, years ago, we did a trauma, it was for a county department of mental health. We were looking at sort of trauma, and we were using existing measures that were validated, but sort of putting them together into sort of a screener. And we were trying to get at trauma. And one of the first questions were some of those that were pulled from the ACEs. Have you ever, before this case, the age of 13, were you hit, punched, kicked, abused by an adult that you trusted? And it was literally the second question after like name, because we only had 5 questions that we were allowed to ask for this sort of project. And people were just like, no, 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 no. People just didn't want to go there because we asked too much too soon. So thinking about how to order your survey is very important. And we also want to, we pre-test everything. If we're designing it ourselves, we want to pre-test it. We've gone into the field with very, very big surveys and had way too much content, doing them on a computer where there was a lot of skip patterns. And we realized that there were way too many skip patterns. It was way confusing. There was way too much that we were asking. And we only learned that through a very rigorous pilot. That really helped us refine the instrument better. So we kind of think about within these surveys that sometimes we have what we call composite measures that will, within a survey, there'll be a scale. You guys saw some of those scales like disagree, strongly disagree, disagree, all the way up to strongly agree. Those measure intensity, scales measure intensity within a survey. And indexes like the ACEs just give you all these questions and they give you 1 score. And out of all of these questions, they produce 1 score that tells you something about a person. So in the cases of the ACEs, having the score of 4 means that you've had a lot of exposure to childhood trauma. And we can take that score and we try to look at how it interacts with risk later in sort of people's healthcare and in terms of health disparities. And so sometimes within a broader survey, we kind of have these sort of smaller subsets that we can analyze as well. So here's an example of a scale. And again, here is an example of the ACEs, so an index. And if your score is 4 or more trauma exposure when you were young, you have that much more, with each additional exposure to trauma as a youth, you have that much more likelihood of later developing an alcohol use disorder. Okay, so these are why sometimes even within a survey, we can have some sort of smaller like scales or indexes that help in our research. Getting at how the modes of administration. So this is, are we doing this, as I mentioned, are we mailing it? Are we doing it online? Are we doing it on computer? Those are some self-administered ways. And sometimes we do these in person, whether it's over the phone or over a telephone. And research shows that these produce, first of all, we get very different response rates. And the goal should be to get a lot of responses, but there's a lot of challenges to survey research and how we do it and what the response is going to be. So something that's self-administered isn't very costly. You can toss stuff into the mail, you can do an internet search, an internet survey, and they offer a lot of anonymity and sort of avoid that interviewer bias that can happen. But response rates are notoriously very, very low. We have no control who completes it. Web-based surveys are, they're the easiest, but they're by far have the worst response rate. And we have very little verification of who's completing it. And so there's lots of design issues with those. If we do interviews that are face-to-face, we often get less missing data and we get a lot higher response rates, but it's also more expensive. You have to hire research staff to be able to do this. Most of us who have been in research for a long time have been interviewers, and we've collected research in the field. And it's a lot more work and it's more intense. You have to train people to do this properly. And there's also biases that happen. I put here an example: appearance, tone, and dynamic between interviewer and interviewee. And I said hot girl, hot guy. you know, we used to have an interviewer that would always be like, oh, but he's so cute. I hate asking all those questions. They're so personal. And he's like, you know, whenever I come across someone, you know, there's real dynamics that happen with real people when you're doing work face-to-face that doesn't happen in these more anonymous types of settings. And that brings both good and bad, this sort of interviewer effect. We did some research that was looking at the presence of an interviewer with a screening tool. So we were looking at screening for alcohol and substances within various settings. And this has been replicated a few times. One of the projects that we did was in Vietnam. We were looking at whether or not it was done on a screening tool, was done for risk on a computer or tablet, and what people said their risk behaviors were. And then we did the same thing, but in face-to-face. And we found that whoever filled out the tablet universally was riskier. And people who were getting the same risk assessment face-to-face, they were much lower risk. And since these folks were randomly assigned, you would think, wow, well, maybe this was just sort of random. But we actually did something similar here in the health study, in the health department, in the student health center, where we did some risk assessments and some screening on a computer or a tablet. It was like a kiosk, and some people were randomly assigned to get the same screening in person. And again, we found that universally people who filled out this screening sort of anonymously were much higher risk. So again, there is this thought that sometimes doing work face-to-face, people are likely to minimize some of their risk when there's like an interviewer right in front of them. And we know that a lot of research shows that females tend to really underreport sexual partners, whereas men tend to overreport sexual partners. Kind of makes sense, right? And so we don't, there's sort of this balance that we have to strike in terms of how we're administering our questions and kind of acknowledging those biases in the process of doing survey research. Again, there's training of interviews. If you're doing stuff that's face-to-face, there's going to be training involved in how you can train your interviewers to be non-judgmental, to not reveal feelings and thoughts, and particularly in the type of work we do, where we're asking a lot of questions about people's kind of private behaviors, things they don't always like to share with people, whether it's sexual experiences, whether it's drug use experience or risk-taking, or maybe it's family trauma. There's a lot of reasons why people have stigma and shame around the things that they're talking about, and we're asking really sensitive questions. And so training your field staff is super important if you're going to be doing stuff that's face-to-face. And again, there's training interviewers, and there's also a lot of supervision that happens to ensure that the methodology is standard in the field. And of course, there's telephone surveys, which are our very least favorite, super unreliable as well, but it happens and people do it. The random digit dialing, there's lots of bias in whether somebody uses cell phones versus landlines, and the big age differences in that. Hardly anyone has a landline anymore, except for oldies like me. I still like just to have one just in case, even though the only people who ever call me are people looking for money or asking questions. So there are some real biases in telephone surveys, but they are a little bit easier to control and to sort of monitor when all of your interviewers are in one location and collecting data sort of on the phone in one location. So there's lots of different strengths and weaknesses to surveys themselves. They tend to be the most popular in social research because we can get a lot of information from if we do our sampling well, we can get a lot of really good information from a relatively small sample. And so they make large sort of studies more feasible. And there's a uniformity and a kind of cleanness and a standardization to a survey research design, where qualitative methods can get really rich and you get a lot more details. And sometimes it's very kind of interesting and exciting, and you can learn a lot about certain patterns, but there's not a uniformity to it at all. So we don't know, some cases are anomalous, maybe they're unique or distinct. And so it's hard for us to draw larger patterns from that type of work. Whereas survey, we can draw some pretty broad patterns, and there's a lot of generalizability to using surveys. But not everybody fits into the, you know, the round hole, the square peg into the round hole. It is real with surveys. You've probably completed surveys yourselves at some point where you've said, I don't know, it's not exactly the right fit, but I guess I'll just put this. And that happens a lot in survey research, is that we're not always, it doesn't really capture the whole of social life or context, but it gives us a flavor. And certainly with some self-report, there's that artificiality about it. And so those are some sort of the strengths and weaknesses here. All right. Let's see if you guys have any questions for me. I'm giving you 15 minutes of your life back, because I know Dr. Shabta comes on in about 15 minutes, but unmute and ask any questions if you're interested.
Hi, Dr. Clarkins. I don't know if you recognize me, Robert,
Hi,
with the LA Trust.
Yeah.
I had a couple of questions. One is, how are surveys being redesigned in the age of Facebook analytics? So now you could actually look for people, look for samples that look like the, you know, people that you want. But obviously there's a lot of ethical and kind of,
right.
Is this a real person?
Yeah. You know, it's a good question. I'm not doing a lot of that work, so I'm not the best person to sort of answer, like, how are we getting who we think we're getting? These are all the questions. Are we getting who we think we're getting? There's also, well, surveys, online stuff has a lot of anonymity, which can be, it can be beneficial. People sometimes are willing to share things that they're not sort of face to face, but that anonymity also brings with this a sort of like potential for a lot of falsification. And sometimes people are less authentic. And I think discerning that has been pretty tough. I think generally in the work that at least I'm involved in, we are using some of that online stuff as maybe sort of one component, but I don't know that people are hanging their hat on that because of the scientific, lack of scientific rigor or knowledge that we have about that. I don't think that anybody is, no one's going to be publishing in the top level journals with just that kind of work. Yeah.
Thank you. And then my second question real quick was, I don't think they're scientific, but a lot of these surveys include an other, where you could include, you know, type in some information. And obviously that sometimes lends itself to add some anecdotal information that might not be answered by question. Is that considered scientific? Is that kind of just a survey for sake of surveying?
That's a great question. What we often do is, so if you are designing your survey in a way where you have other, and sometimes we build them where we have our close-ended, our things that are closed-ended, and there's a forced response set. But a lot of times we have some open-ended questions, and those require kind of qualitative analysis. So a lot of times we start to code themes from there. So depending on what the topic is, we will pull all of those open-ended responses into files and we'll start analyzing them qualitatively. So we'll start doing a first open coding where we just kind of go through and see what are the big themes, what's the general big patterns that people are saying. And then from there, we start doing sort of a sort of secondary coding where we start to kind of do a little deeper analysis, but also pick out some of the money quotes, the ones that really say something valuable and important. The one kind of problem with, I don't know if I would say problem, but when people get in a survey mindset, they're not always as open to really responding very much. So a lot of times we have to try to encourage responses in those open. People tend to put, you know, one sentence or 3 words, incomplete sentences. So the value, at least in my experience, has been kind of limited unless we really present this as a mixed methods. Like here's some quantitative stuff, but we also want to do some more open-ended where you can share and explain more. And it's not always captured well in that other, but sometimes we are able to quantify the kind of qualitative information. So sometimes, you know, people will put 1 or 2 words and we're able to sort of quantify that as well and include that. So
Thank you. Yeah, thank you.
Great, thank you. Great, well, if there's no other questions, I totally appreciate everyone's time, and I hope you guys have a great training series, and always interested in hearing the type of substance use research that you're working on, all the new smart kids in the field, pushing us oldies out. I love it, it's great. So good luck in your research, everyone, and thank you.
Great, thank you so much, Dr. Larkins. I know you can think about this, you don't have to go on the spot, but would you be willing to be a mentor for some of our trainees?
Absolutely, absolutely.
Okay. Great, thank you so much. And I think Lewis, who asked the question, might be a good person to have you mentor him. Really
Great,
good.
absolutely.
Do we have anyone else have any questions?
I had a question. What is the standard process of publishing your survey? In other words, how do you go from creating a survey to making it accessible to other researchers? Is it similar to creating an article where there's reviewers, or how does that happen?
Okay, so you're saying if you are, you create a survey and you use it in your research?
Yes.
Right, yeah, good. So this does happen a lot where, you know, maybe we were working with a project with RAND many years ago, and there were a lot of different sites, and we wanted to have a uniform sort of instrument, and within we had a bunch of smaller validated ones, but everybody had their own kinds of questions that they wanted. We were creating kind of something new. And in the process of, I think in this process, sometimes you don't find something that's perfect for your needs, like whether your target population is slightly off, or the instrument is really targeting, you know, I did some stuff like ethnic acculturation stuff, and it was like, oh, well, it's really targeting more African-American and Latino communities, but there's not anything similar for sort of Asian communities. When this was years ago when I was looking. So can I adapt or modify existing tools? And that does sometimes happen, where we've sometimes gone to other authors and asked to kind of use some of their stuff with adaptations. And sometimes you create something new, and then from there, you actually, as you start publishing, people will start to come to you. And, you know, there is a process by which, you know, you validate an instrument and it has psychometric properties, and then it becomes sort of more widely available to the public. But I do think that that this does happen, where there's something out there that doesn't exactly meet your needs, and you start to go down the path of instrumentation development. And it's not a, I would say it's not an easy process, because there is a lot of sort of statistical work that goes into this. Years ago, I was working on an instrument called the DD-CAT, or the DD-MUT, which is a co-occurring capability. It's a site, it's specific to sites, and it's on co-occurring capability. How capable is your substance abuse treatment site, your primary care site, or your mental health site? How well does it treat people, or how responsive it is to people with co-occurring disorders? And I know that the developer of that started out with using different things and then realized nothing quite fit, so he sort of developed his own. And he would always say, the process of developing your own instruments is not one I would suggest, because it's, you know, it's a lot of work. But he was really able to build a sort of compendium of instruments that were incredibly useful to the field, because there was nothing else out there that really helped people understand how dual or multiple disorders, how well facilities are able to provide integrated care. And so it does happen that you might not find what you need, and you're willing to develop your own. Yeah.
Thank you.
Also a question, do any HIPAA rules apply to the surveys, or usually not because they're more anonymous?
Well, it depends. Yes, they can. So it depends if you're doing like a survey that's more in a treatment setting that addresses substance use. If you're doing like clinical treatment populations, which we've done before, you would be, you need to follow 42 CFR, which protects substance using populations, which is different than HIPAA. But if you're collecting data, this would all be as you go through the process of through your IRB. If you have stuff that's completely anonymous and it's all de-identified, there's no way of tracking people, and your distribution of where you're disseminating your service or collecting your data, that would probably get cleared through your institutional review board as exempt, and there would be certain rules that would not be applicable. But if there is any possibility of, and you're collecting in say healthcare settings or stuff like this, all HIPAA and 42 CFR rules are going to apply. And you would have to make sure that, you know, you had IRB approval before you started asking any of these questions. You'd have to go through the formal scientific research process of working with human subjects. Great, good question. All right, thank you everyone. And I'm happy to mentor folks or anybody who's interested in, I teach research methods here at UCLA for undergraduates, and always happy to help in that process. So.
Great, that was a great talk.
Thanks everyone.
Thank you so much. So people can have a couple-minute break till Dr. Shoptaw comes on.
I gave you 5 minutes of your life back. Isn't that great?
Right, very generous.
Thanks, everyone. Bye.
Bye. Thank you.
So I'm here, Theodore.
Hi, welcome.
Hi, Steve. Welcome. Thank you for joining us. Do you want to pull up your slides?
Yeah, let me just do that real quick and make sure that they show up.
I'm actually going to start by sharing my screen to go over the CME information.
Oh, there you are.
Okay.
Hi, Bonnie. Good to see you. I didn't see you. I was looking for you on the screen. I saw your message and I couldn't find you to make you the co-host. But
oh,
okay,
sorry.
Bonnie.
I'm here. Yeah.
No, that's okay. Thank you. Great. You should be able to right now.
Okay, thanks.
You're welcome.
And then just go to the presenter view.
Yeah, it's not working. I'm going to have to unshare and do that. Hang on just a second.
Looks like you might be in presenters view.
Okay, does that look okay?
Perhaps if you'd like to go to display settings up in the top, because right now you're in presenter view.
Okay.
So if you click on display settings at the top.
Okay.
We just had this with the other presenter.
Yeah,
so I think we figured it out.
Here we go. Nope. It's weird that it's not working.
Did everybody? Oh, there it is.
There we go.
Is that it? Okay.
No, so go. No, it still has the notes on the side. So go to display settings at the top.
Mm-hmm.
See where it says show taskbar, display settings, and end slideshow? The middle one, display settings.
Huh. It's not showing me that. That's very weird. Hang on, let me unshare. I haven't had this issue before. Let me see here.
Yeah, we had it, I think, with our last presenter.
Okay.
It might be a Zoom thing today.
Yeah. It's like messing
If it's
up my,
readable in the other format, just go.
yeah, it's messing up my whole desktop now, which is odd.
Oh no.
Let me see.
It's good. Well,
Okay.
you still have the things on the left side.
Yeah,
Yeah,
it's not. Whenever I go
whole-
to presenter mode, it messes up the whole screen. So
Well, maybe you can just go to slideshow, see at the top.
Yeah,
how there's-
right. I think go to slideshow, show it will be fine.
And then like see where there's like home, insert,
no,
design,
no,
transitions.
Yeah, just do that slideshow down at the corner there. See what
Yeah.
happens with that.
Yeah, that's the one.
Yeah.
Okay.
yeah,
Right.
is that better?
How come?
Yeah, it still shows the presenter view. And you don't see display settings at the top?
Okay, display settings.
Oh, there you go. Okay, and then and then swap presenter. There we go. Yep.
Okay.
And then that.
never
Yep.
There we
Perfect.
go. Okay.
This is good.
Sounds good. All right, give me just a second here.
And if you're not muted, could you please mute yourself? Thank you.
Okay, we'll go ahead and get started. Thank you everyone for joining us for today's Friday Noon Lecture Series presentation with Dr. Shapta. We'd like to begin with a few notes. By default, participants' video is turned off and all participants are muted. We ask that you keep your video off and remain muted for the duration of the initial presentation. We will have Q&A at the end. If you have any technical difficulties, please message the Zoom meeting host. We would like to inform you that this session will be recorded. Today's session is accredited for CME credit. And these are the overall learning objectives for the Friday Noon Lecture Series. These are today's session-specific learning objectives. None of the individuals involved in this activity had anything to disclose. Charles R. Drew University is accredited by the Accreditation Council for Continuing Medical Education to provide continuing medical education for physicians. Charles R. Drew University designates this live activity for a maximum of 1 AMA PRA Category 1 credit. Physicians should claim only the credit commensurate with the extent of their participation in the activity. In order to receive a certificate of credit or certificate of participation in this activity, the evaluation and application for credit form that will be posted in the chat at the end of this session must be completed. Certificates will be emailed to you on a quarterly basis. If you have any questions, please contact the Office of CME. That wraps up the CME accreditation information. So I'll now turn it over to Dr. Friedman to introduce today's speaker while we transfer the screen share.
Thank you, Bonnie. Thank you for sponsoring the program, for providing CME for the program today. Dr. Steve Shapta is a good friend of mine. He's also a clinical psychologist, professor and vice chair of research in the Department of Family Medicine at UCLA. He is a professor in the Department of Psychiatry at UCLA, and I didn't know this, at the University of Cape Town in South Africa. Dr. Shapta's research involves developing medication and behavioral interventions for stimulant and opiate use disorder, especially in the context of transmission of HIV. He directs 2 centers at UCLA, the Center for HIV Identification, Prevention, and Treatment Services, and the Center for Behavioral and Addiction Medicine. He recently became the multiple PI for NIDA's clinical trial network, Big South/West Node. Dr. Shapta is very involved in, and has been for years, in the drug abuse research that we're doing at Charles Drew. And we're delighted to have him be the keynote speaker of today's first Research Institute. Thank you, Dr. Shapta.
Well, thank you, Theodore. It's awesome to be here. You can hear me okay?
Yeah.
Good. And it's always a tough slot to follow Sherry Larkins. So I'll try to do my best. This could be a really crusty, boring talk, but I hope that I'll be able to keep it full of life for you, because this is my life and I actually kind of find it exciting. So we'll be talking today about designing and implementing randomized clinical trials in drug addiction research. And with that, we'll start talking. We talked a little bit about my funders. Thank you, Dr. Friedman, for doing that. And also to point out that periodically I'm running trials, and I do get clinical supplies in the past 3 years from Gilead Sciences and Alchemies, but none of that rises to the level of a conflict. So the objectives for today is to talk about the question. What is the question? We're going to talk a lot about the question, because sometimes the question gets lost in the design and the research activity. The most important part of the research work that we do here is about the question, and that how you ask the question greatly impacts your design choices. We're going to talk a little bit about phases of medication development. It's particularly important now, particularly not only for addiction science, but also for vaccine science. And some of the concepts will map onto the development of the vaccine trials. And then stages of behavior therapy development, and how these stages and choices and questions affect the design. And we're going to end with a focus on primary endpoints. We're going to talk a little bit about equity, diversity and inclusion, and we're going to talk about how internal and external validity, particularly around these concepts, are in play. But first, I want to start talking about what is the question. First things first, what is addiction? We're going to talk about inclusion and exclusion criteria, and causation or lack of causation. So I apologize that I don't know you all, so I don't know where your understanding is about addiction, but addiction is a spectrum disorder. It ranges from use that doesn't cause problems, or as my friend Kathy Reback likes to say, it's fun, to its use that causes occasional problems, occasionally to frequently. And as you can see, as we move across from the left to the right, you start having a smaller part of the triangle. It means fewer people will continue using the drug because that's what happens. Nature tells you when something is not good for you, you stop it. So fun becomes fun with problems. And then as you move further to the right, and you start moving into mild or moderate substance use disorder or addiction, you have fun with problems just becoming problems. So here you have just people at the far end of the right curve here. You have fewer of them, but they have more severe problems. At that point, it's not about the function of the drug. So you might have started using heroin or cocaine or caffeine or whatever it might have been for the reasons that there could be any number of reasons. But when you hit the issue of severe substance use disorder and addiction, you're really talking about just drugs being used because the brain has gone through this process of neural adaptation, and you have addiction and problems. It's important, I encourage each of you to pull down the DSM-5 definition about stimulant use, about substance use disorder. Yeah. This is really important. That the substance use disorder across all the substances is a maladaptive pattern of use with clinically significant impairment or distress. That's in italics, because if you don't have impairment or distress, you can have people using the same amount of substance and the same maladaptive pattern of use, but they wouldn't qualify for the diagnosis. An important issue. And 2 or more of the following in the same 12-month period. I'm not going to read through all of these. You can do these, but on your own. But the first 2 are the only 2 that are really tied to biological systems. One is tolerance, that you need more of the drug to get the effect you wanted when you first started using the drug. So that's like you need a bottle of wine as opposed to when you first started drinking, you needed a glass of wine, or you need, you know, a 12-pack of beer when you first started, two beers got you really drunk. Withdrawal, when you stop using the substance, you actually feel something bad. A hangover is a withdrawal symptom. Opioids and stimulants, many of the substances have withdrawal symptoms. You continue to use the substance for longer periods than intended. You can't cut down or quit. Times you can read through here, but I want to focus on item number 8. It's continued use despite knowledge of negative consequences. This is important when studying people with addiction because this is the major defining criteria of addiction. It's the continued use despite knowledge of negative consequences. My mother, who's dying of stage 4 lung cancer, continues to smoke. There are days when she understands that her smoking behavior is what caused this stage 4 lung cancer. But most of the time she's walking around talking about how it was exposure to workplace pathogens that caused these tumors in her lungs. So I'm always struck with, is my mother that dumb that she doesn't understand this? The answer to that is no, she understands this. But it's about the way in which the brain gets reorganized to understand how their behavior around substance use is not necessarily tied directly to negative consequences. And the idea that this other one number here of number 4, you can't cut down or quit. It's not like you haven't tried or you can't figure this out. It's just that you've got addiction and you can't cut down or quit. This is a fundamental important point. I spent time on this. I want you to meditate on this. When I say I meditate on item number 8, I actually do. As I drive around town, I think about how people continue to use substance despite their knowledge of negative consequences. It's a whole lecture. It's a whole lecture series I have of the sequela from that item. But it's really important in terms of doing research with people with addiction problems. What is often comorbid with addiction are mental health disorders, and recent work is thinking about these mental health disorders as a comorbid spectrum. This is a very busy slide, but again, I want to emphasize this severity spectrum here, where you have a lot of folks who may be anxious and depressed. Over here, that doesn't cause much dysfunction, whereas you get closer to the end of the triangle and you have fewer people, but their disorder causes much more impairment. You can see the same sort of thing in the clinical syndromes of mental retardation, autism, schizophrenia, schizoaffective disorder, and so forth. And you can see here across the symptoms of mental health disorders, the reasons and the causes, you can see all this coming together. But the idea here is you have this spectrum disorder as well that ranges from mild or no problems all the way to severe. So what does that mean in terms of design considerations for addiction trials? Well, let's think about it in several different ways. One is you can have observational approaches. These are very large approaches where you might be counting the density of addiction-related phenomena. For example, alcohol retailers, cigarette ads, or discarded cigarette butts, or discarded needles in neighborhoods. Those are things that don't need IRB approval, but that actually tell you something about the substance happening within the particular phenomenon. Measuring drug or drug metabolite in wastewater, a very novel new way to understand how much substance is being used in a metropolitan area. Very fascinating, and it gives you a sense of what's going on. That, there's a lot of discussions and talking about observational approaches, but this idea now about mixed methods, Sherry may have been talking a little bit about that, where you have, say, an observational approach integrated with a clinical trial, gives you even more better ability to have external validity because you understand what's happening around the neighborhood where your study is happening. Epidemiological approaches. These are large surveys, often that have representational sampling strategies like household samples or time location samples that tell us about where people are in terms of their substance use across the population. And then you have, related to that, you have targeted sampling strategies that use convenience or snowball sampling to be able to get more to the question. So the question about how much or what you want to know is important. If you want to know how much substance is being used by people who have stimulant use disorder, you don't want to do a household sample. You want to do something like a convenience or a snowball sampling because it's much more efficient to get the information. What you gain in efficiency, though, you'll lose in external validity. because you have this convenient sample, you're not able to extrapolate findings. And then finally, thinking about efficacy and effectiveness approaches, we look at things like behavioral pharmacology. These are very controlled settings where we ask the question of safety. Small initial trials where we continue to explore safety and then have some understanding of efficacy. Large trials where we talk about efficacy and effectiveness. These are different things. Efficacy is the effect of an intervention compared to a control. Effectiveness is measuring the impact of a treatment on a population. Those are two separate things and require two different kinds of designs. Community level trials are ones that talk about effectiveness. This is not a place where you would do an efficacy trial. So you can see how that the question you're interested in asking is going to directly affect the question, your design choices. We talked a little bit about external validity, and I want to focus here on inclusion and exclusion criteria. If you're doing a trial, one of the things you want to be able to do is to say that the people who you're including in your trial or your research are a particular kind of general population, which is widely inclusive, or in a clinical trial where you're going to be focusing on a particular group of people who use substances. So that's what you will do. I want to point out something here in that first bullet. Most people who use drugs, and I put that paren in there about person-forward language. Because we study addiction these days, I'm particularly sensitive to using person-forward language. We don't talk about cocaine addicts. We don't talk about cocaine dependent people. We talk about, well, actually we do talk about people with cocaine dependence or cocaine addiction. But if you end the term with people, it becomes a stigmatizing point. So we want to use person-forward language. But most people who are using drugs are polysubstance users. They're combining many drugs. It could be one or two extra, but especially with the increase of cannabis in the population, it's really hard to find pure groups of cocaine users or people who use methamphetamine or people who use marijuana. People often will use multiple substances, and it becomes important then for you to figure out what level of other substance use is acceptable for your question and your study design. Mental health disorders, same sort of thing. These frequently co-occur with people who use drugs. And then you want to have to define the type and level of mental health conditions that you want to bring in. If you're doing a clinical trial on an antidepressant medication, you definitely don't want to include people who may be using cocaine at a very high level, but who are also depressed and on antidepressants. Finally, looking at questions about how people who live with addiction face barriers to structural determinants of health. And this includes things like housing instability, the lack of ability to store medications when you're living close to the street. It can be very hard to include people who have unstable housing. I've done it, but it increases a level of complexity into your design and how you have to work with your participants. And it also interferes or jeopardizes gains that you make from interventions. If you're living close to the street, it's very hard to maintain gains in your health regarding substance use or mental health if you're periodically being rolled out of your housing situation. Lack of healthcare and medical instability complicate everything. And poverty. Poverty affects the ability to remain in the trial and provide follow-up visits. And so the questions about how you want to address these very important external validity questions is key right up front. So when I often give a talk about design, people ask me, what tests should I use? And that always comes back to the issue of what is your question? The question of the data is far more important than the data that you're going to be collecting. So in general, I like to refer to Tabachnik and Fidel's classic using multivariate statistics. I know there's some biostats people on the call that may have other recommendations, but they have a chart that's really helpful for simple people like me. And basically when you're analyzing data, there's a difference between groups, and there's a set of questions, a set of options you have in statistics. to be able to answer those questions. T-tests, ANOVAs, MANCOVAs, MANOVAs. There's different kinds of ways that, you know, if you've randomized a person to a group or a control, that you'd be able to test the question between those two groups. Often those two groups, so those are in experimental questions, but also in quasi-experimental research, where you're looking at an effect of, say, trauma on people who use substance, and being able to compare a group who had that experience versus a group who didn't. It's not randomization, it's quasi-experimental, but again, we're comparing two different groups. In terms of looking at correlations between variables, this is the flip side of the coin of comparing between groups, but it's basically recognizing that you have one data set, you have a sample, a survey, like Sherry was talking about, a survey or a household sample. You have one pool of data, however big it is, and within that data, you're going to be doing things like multiple regressions, or you're going to run linear models, or you're going to be able to test the association between those variables. In the differences between groups, you have better ability to have a statement about efficacy, with correlations between especially if you have randomization going on. In the correlations between variables, you really are limited in what you can talk about in terms of effect between variables. You can talk about linkages or correlations, but the cause is something you can't really get to very well. And then a third approach here is the latent structure, or looking for underlying relationships between variables. And these are things like cluster or factor analysis and structural equation modeling. The bottom line of this slide is to say that what's important is your question. How you ask the question and the sample that you pull together will determine the statistical, will largely advise the statistical approach. So I want to start with asking this question here. So what is the best design for this? You want to know if cigarette smokers who have a genetic polymorphism for rapid nicotine metabolism, there is such a thing, it's expressed as a ratio, and it's the nicotine metabolism ratio. If they benefit from nicotine replacement therapy in the same way that persons who don't have this NMR, what is the most efficient study design you would use to answer this question? Is it 1, design observing outcomes for nicotine replacement therapy in smoking cessation clinics? Number 2, is it design testing population prevalence of NMR and correlating smoking status? Is it 3, behavioral pharmacology design, testing nicotine, cotinine blood levels of smokers with and without NMR exposed to cigarette smoke? Or is it 4, an open label trial design of outcomes for subjects with and without NMR of nicotine replacement for smoking cessation? Okay, take a second and kind of consider these options. And since we're on Zoom, because unfortunately when I like to ask, I like interactions. It's kind of hard to have interactions. Let's walk through these.
People can
the best
answer
here
if
is
they want also.
Okay. Anybody want to take a stab? I'll walk us through this. So the best answer here is 4, the open label trial design of outcomes for subjects with and without NMR. The reason for that is we want to ask the question, do people with this genetic polymorphism benefit from nicotine replacement therapy in the same way as people who do not? So even though an open label trial doesn't let you test against a standard, it does let you test whether NMR outcomes, people who have NMR or don't have NMR, have different kinds of responses to nicotine replacement smoking. You can make an argument for 1, 2, or 3, but the most efficient study design here is number 4. So we can talk about this more later if you want in question and answer, but I've got more questions that I'll keep your interest going here. So in terms of designing, so let me stop here for questions.
We don't have any in the chat box, but if anyone wants to unmute and ask the question, they can.
Okay, so we're going to move on then. So I want to focus on addiction research and talk about therapy or intervention development and stages and why they matter, medications, behavioral therapies, and phases. So I'm going to go back to my point that I started the talk with today, and that is, what is the question? There are very few medications for treating addictions, and the reason is because we ask the wrong questions. We don't ask a question, what should a medication do? We actually just go in and say, can we reverse a symptom of addiction, which in certain ways we can. So in terms of targets for pharmacotherapies, one is an agonist approach. It's a way in which we have medications that can function in a similar way that the drug that people are abusing. So methadone, buprenorphine are two agonist approaches that work in similar ways that medications for opioid, as does heroin. So does nicotine. Nicotine is the same drug that you get out of a cigarette. So it actually helps people to not use the drug of abuse. And the critique of this approach is you're swapping a drug for a drug. I don't believe that's true, but we can have that discussion later down the road. The antagonist approach, the medications that work in the opposite direction to the drug. So for opioids, it would be things like naltrexone or naloxone, both of which work in opposite direction to heroin and the opioid agonists. Drugs that relieve withdrawal symptoms, things like clonidine for opioids or lisdexamfetamine for stimulants. These are drugs that actually help people feel better when they're stopping their drug abuse. Stabilizing mood. Some people think that naltrexone stabilizes mood by blocking some of the euphoria that happens when you use a drug. That's one of the reasons why they think naltrexone works for alcohol use disorder. That when people get buzzed from drinking, when you're on naltrexone, a good number of people in the population don't feel that buzz. They just get sloppy drunk. So they stop drinking as much. And then finally, the question about long-acting formulations. As we get medications on board, we can that have efficacy, we can start thinking about, well, maybe there's ways that we can take these medications and formulate them in ways so that they're on board for a long period of time. So people don't have to always be asking the question about whether they should be in recovery or not, because they have a medication on board that helps them to meet their substance use goals. An important piece of trial design in working with addiction medications and behavior therapies, but particularly for medications, is the consideration of the ethical use of placebo. At this point, we have this directly affects design for smoking studies or opioid medication studies. As we have medications that are effective for smoking or opioids, it's now considered unethical to use a placebo-controlled design for a new medication for stopping smoking cessation, or for treating opioid use disorder. Whatever the medications might be, we have effective treatments. And so what we need to ask the question now is whether a potential medication that may be new could actually improve over or be equivalent to efficacy of a standard effective smoking cessation or opioid use disorder treatment. And then finally, back to that point about we don't ask the right questions. None of these treatments can cure addiction, behavioral or pharmacological. Addiction is a chronic relapsing disorder, and multiple treatments are usually just required before abstinence is achieved over the long term. So that means that as people, we're walking around with addictions that affect us over our lifespan. Dr. Grella talks about the career, the addiction career, but the treatment that we have is for a point in time or several weeks in time or for several months in time, as opposed to a lifetime. So the idea here about asking the right question is important. I think this has really impaired our field in terms of not paying attention to this. And it's something that a budding career scientist should focus on, I think. I certainly would if I was starting my career again. In terms of behavior therapy trials, the targets there are several of them, and these are important. So one of the one of the questions is, can you give me something that helps me stop using drug? Can you help me stop and instill abstinence? That's a behavioral target. I can do that. What about once I stop using drug and I lapse or relapse to drug, can you actually give me something behavioral that helps me to prevent that lapse or relapse? Yes, I can do that. What about improving mood, craving, or overall functioning? Yes, we can do that with behavior therapy. And what about improving cognition or motivation? The answer is yes, we can do that as well. Again, the issue of the importance of placebo control. We'll go into some of the specific therapies in this in a second, but I wanted to just kind of give you a background here of the palette of medications that are approved for use. You can see over here, there are no medications on the shelf for cocaine, methamphetamine, or cannabis. So that out of the way, we can see that for smoking cessation, there are 3 treatments, rank order in terms of efficacy, with higher efficacy down here: nicotine replacement therapy, bupropion, and varenicline. These are the smoking cessation medications, and they increase in efficacy. If you combine them, you get better outcomes than a single medication by itself. Opioids, the full agonist of methadone, the partial agonist of buprenorphine, plus or minus naloxone tab. It's delivered sublingually. Very exciting new developments are about long-acting injectables that can be delivered in a week or a month-long form, and a long-acting implant that's 6 months long. And then the backside of this is naltrexone, the antagonist, which is can be oral or injectable. For withdrawal, we have clonidine, and we have 4 medications for alcohol use disorder. I want to just kind of hammer this point home. There is no reason that we should ask the question, can we help our person who has opioid use disorder to become drug-free? That is a wrong question to ask. One of the contributions to the ongoing opioid epidemic right now is that people detox or go into some other treatment program to become drug-free, and then relapse either often in the parking lot on the way out of the treatment center. When they load their dose that they're used to taking, because they've lost their tolerance. Remember, tolerance, that's there. You need more of the drug to get the same effect. So if you go and you load your normal dose before you went through your treatment, often people overdose and die. So what we like to have on board for the question we like to ask for people who have opioid use disorder is how can we help you best get your life back? And that means being able to go to work, have a family, pay your bills. And that usually means not drug-free status, but some sort of medication status, whether it's buprenorphine or methadone, that lets people move back in their life and stay active, okay? On the psychology side, the importance of addiction is that the disorder from a psychological perspective is about impulse inhibition. Patients will tell me often that, Doc, it's like my cognitive brakes are shot. In my brain, there is no brake pedal. It's all gas pedal. That is the exact psychology of addiction. Why am I saying this? Because it's important to understand this to ask the right question. So we're doing things now to talk about ways in which treatments might change this impulse inhibition problem, to be able to build some way to break or slow down the individual so that they can make decisions different from actually using drug. The key factor is ambivalence. The underlying fulcrum here is that I have a problem and I don't have a problem. 2 thoughts being held at the same time. They may be more or less higher in strength at any different time, but the important part about this is the ambivalence is never resolved. I've talked to smokers who've been smoke-free for 30 years, and they will still say, I would sure love to have a cigarette. And they follow it up with, but I know that if I have that one, it'll be a pack. And if I have a pack, it'll be a carton. And if it's a carton, I'm back on the wheel. I need to not do that. Lots of comorbidities. We talked about that already. And then how do you know your patient is lying Well, one of the answers is their lips are moving, because one of the hallmarks of addiction is being able to do whatever you need to do to get drug or to use drug. So what we, instead of using outcomes that focus on self-report, we're going to use biomarkers so that we don't have to rely on our patient who may come in thinking that I need to feel good about myself. I want my therapist or my counselor or my doctor to actually believe I'm a good guy. So I'm going to tell him a version of the truth that I think he'd best like to hear. So that often happens. It's not fair to put that onto our participants or our patients. That's a function, a symptom of addiction. And the best way to manage that is to just use biomarkers, taking urine samples, taking a breath sample, taking a saliva sample, some sample of the body that tells us a little bit with more certainty about exposure and use of a substance. So we talked a little bit about this before, that there are behavior therapies to instill abstinence contingency management, providing biological financial reinforcers for biological markers of abstinence. The most effective treatment we have out there is contingency management. I'm sure most of you on the call don't know anything about it, so I don't want to spend time on it. But if you want to read about it, it's really cool. It's been a major part of my career, and I believe in it. It's coming to a treatment center near you because we're making change and being able to bring this to the communities, but it is very potent. Motivational interviewing, the ability to talk with people and find out what are the pros and the cons of a substance to them that are specific and unique. Non-directive, person-centered, very potent. Cognitive behavioral therapy, teaching skills how to instill abstinence and to prevent relapse. And most importantly, how to get back to abstinence once relapse happens. And then finally, 12-step facilitation, which is an effective treatment that actually helps people to get hooked into the 12 steps programs in our communities. So here's our second question. A scientist from South Africa tells you that a new plant-based medication, Sclerotium tortuosum, is a PDE4 inhibitor that causes stimulant effects in rats, conditioned place preference, and reduces anxiety in humans. You decide to evaluate this drug for meth addiction. What target would best be thought of in developing the medication? Would it be to be an antagonist medication as a blocking effect, an agonist medication as a substitution effect? Would it be a withdrawal medication? It reduces withdrawal symptoms. Or would it be a relapse prevention medication? It prevents resumption of active methamphetamine use. So think for a few minutes. Anybody want to take a stab at this one?
Would it be 3 withdrawal medication?
That's a great choice. Absolutely.
Withdrawal medication.
That's a great selection. Yes. And it could also be a relapse, an agonist medication for sure. It could also be a relapse prevention medication, but it would not be a one. It would not be an antagonist medication. So however you set that up would be important. So like if you were to use people and you were using it as an agonist medication, you would want to bring people in who are actively users, randomize them to either the placebo or the drug, and then see how they do. If it's a withdrawal medication, you would bring people in and you would actually have them go through, probably in a hospital, go through controlled withdrawal, and you could provide the medication or placebo and measure symptoms. If you wanted relapse prevention, you could work inpatient, get somebody down to 0 methamphetamine use, and then start the drug or placebo, and then see how long it takes for them, how many days before they resume active methamphetamine use. So 2, 3, or 4, you could actually put together a very strong design to be able to test how that medication might be working. Again, it comes back to the issue of what is the question? How do you want to set this up? Great, great, great. This is awesome. So in terms of a medication, we still have the FDA, even though there's an erosion of many of our administrative functions in government right now. But in terms of how we develop a medication, there's a lot of communication with FDA. It starts before the phase 1 safety study happens, but basically we communicate with FDA and we start phase 1 after we've gotten plenty of data from preclinical animals to be able to say, okay, this medication has a reason to be looked at in terms of addressing addiction. And so we're going to do a first in humans, a few subjects. We're going to do mostly biological measures. If it works through there, we don't kill anybody, we move on to the second question. Let's look at people who might be using methamphetamine and see how it works if we're looking at a methamphetamine drug. How safe is it? Are there any efficacy parameters? We're going to talk to FDA, and then they'll let us go with the phase 2. The phase 2A is the first efficacy studies where we're looking to see how effective the drug is compared to placebo. Smaller studies of 50 to 200. The 2B is the next round, which your studies are going to be 200 to 500. And once that's done, you can file for marketing. The FDA can tell you whether you need another trial or not. Moving into phase 3 with randomized controlled trials and in relaxed controls and inclusion and exclusion criteria and real world studies in clinics. And then phase 4 is the RCTs with samples of greater than 1,000, where we're looking for opportunities to see subgroup analysis. So it's important, one of the direct parallels of thinking here is to think about what's happening with Moderna phase 3 trials, which is they went right into efficacy trials from the first safety study. They just basically skipped phase 2. It's interesting to see that happened. It's because of the medical emergency, but you can see that the lack of experience from phase 1 to phase 3 really does hamper our ability to understand what happens with people with these drugs as they move out of animals preclinical testing, out of the engineering labs and into our communities. So an example here I want to focus on here just to show you, this is a new compound, CS1103. We're working with the company that's developing this. You can see here in non-human primates, this is a molecule that sequesters methamphetamine in urine. It actually sees methamphetamine in blood. It goes to the molecule, it encapsulates it, sequesters it, and then moves it through your kidneys. So it never goes through your liver. It doesn't do any first pass. It just takes the methamphetamine out of your blood. It's an awesome idea for a potential overdose drug and potentially could be brought on board in a long-acting form to be able to help people to stay involved in methamphetamine treatment. But what you can see here is that if you look at the meth in urine after 1 hour in these animals, you can see that it basically eliminates methamphetamine compared to saline. So it's just pretty intense watching the amount of methamphetamine that's eliminated in methamphetamine in urine that's been eliminated. It's just incredible. And if we look over here on the right side, we can see that the effect is dose dependent. So those are two things you want to see in animals. You see that the drug is doing what you expect it to do, and that you can see a dose dependent effect where you see more of the effect with more of the drug, less of the effect with less of the drug, and even so compared to placebo, you're going to see you're going to see more of the effect than placebo. So this is one we may be hearing about more as we get some human experience with this. Our lab is going to be one of the first to actually test this, to test this in phase 1A trials. Here's what we did with ibutylast to teach you what happens with first in human studies, the phase 1A trials, 11 subjects. These are typical studies of 10 to 15, maybe 20 subjects max. Sometimes it can be down to 4 or 5. And the questions here we're asking are, does the drug compared to placebo change heart rate, blood pressure, systolic or diastolic? And does it do anything with different sorts of subjective effects? So we brought people into lab here and we gave them IV infusions of 15 milligrams and 30 milligrams under different levels of different doses of the medication. And you can see that if you look over time, time is getting longer this way, that if we look at the meth at the ratings as they're getting infused, you can see the high dose here is actually showing more of the effects here for any good effects, how high are you, or how any drug effects. This is what you want to see. There's a lot of noise in there because you don't have as many subjects, but it's enough to move forward into a phase 2 trial, which we did recently, just concluded. Keith Heinzerling led this trial of ibutylast compared to placebo and saw that there was no effect in terms of your methamphetamine negative urine screens. And that basically what we saw, that there was no effect specifically for the people who had high rates of methamphetamine use at baseline. But for those who had less methamphetamine use at baseline, we can see that placebo responders did better than the people on the drug. It's too bad. This is a phase 3 study. You can see what this is, the Moderna trial vaccine study. And you can see 30,000 participants. You have huge numbers of people in experimental and placebo condition. And I'm going to start talking a little bit about the primary outcome selections. And you can see it's the number of participants with the first occurrence of COVID-19, with adverse events, with solicited local and systemic adverse reactions. And you can see here that these are the sorts of primary outcomes that get selected. Those are important because you can ask a lot of outcomes to be part of your study, but you need to select the important ones for your primaries. Behavioral therapy development, you can see it follows in the same sort of stage sequential designs where you start with stage 1, which allows people to develop entirely new therapies or to adapt or improve new ones. It uses qualitative methods like what Sherry was talking about to gather information on acceptability, feasibility, and cultural appropriateness. That you have stage 2 efficacy design testing, where you have a standard against the experimental. And then finally, you have the effectiveness, where you're working community-wide to see how well this intervention works to address addiction issues on a community level. To give you an example of this, Kathy Reback, one of the people I've worked with many years, and I think maybe one of your speakers, I don't know, developed a theory-based text messages for reducing stimulant use in gay and bisexual methamphetamine users. She started by developing these texts that you can read from your slide deck, built upon different theories: social support theory, health belief theory, social cognition theory, and was able to show that these texts actually do things. Like what it does is it helps people to reduce their receptive insertive anal sex partners with their main partners, serodiscordant partners, and so forth. And you can see in here measuring just how many messages are necessary to get these sort of outcomes. Again, looking at what's the dose that you need to deliver. To a follow-up study, a phase 2 study, testing efficacy and showing that compared to the text delivered with a person, the text just automatically, and just some health-related texts, you can see that really there's no advantage to adding a human to the texts, that the texts conditions both do better than the standard health control, but that the idea here is you get just about as much bang for your buck in terms of just delivering the text on an SMS machine than you do by having a real human involved. Interesting, important outcome. So you want to develop a new treatment for alcohol use disorder. You're including only people with severe alcohol use disorder to see if it's safe. It's a stage 1 trial. Your patient, so it's a behavior therapy and it's stage 1. Your participants must drink on average more than 15 standard drinks per day. Which of the following treatments is ill-advised for you to incorporate into your new treatment? Is it A, reduce alcohol intake by 100%, complete abstinence? B, start participants on a benzodiazepine. C, begin medically supervised outpatient detoxification. Or D, begin inpatient detoxification. Give you a minute. Anybody
Someone
want to
said
take a
A in
stab
the
at the
chat.
correct answer? Yay! All right, awesome. Why would you not do A?
It's unrealistic.
It's unrealistic, but it's also dangerous. When you take people who are who have severe alcohol use disorder and their brains are used to getting a lot of alcohol and those neurons are bathed in alcohol regularly, and you shut off the alcohol use, what happens is irregular firing. They get mad, they get angry. It causes seizures. And people seize actually and can die from going from a high rate of alcohol drinking every day to 0. So what you would do then instead, if you're doing a behavior therapy, is you would either start them on a benzodiazepine, you'd start a medically supervised outpatient detox, or you'd begin inpatient hospitalization. Another thing that I've used with patients is to reduce their average daily intake by half and let that step down happen every other day, so that you actually wean the neurons off of their accustomed dose of alcohol. But you can kill people doing this. So that's one of the reasons why I wanted to put this in here. So like if you do want to develop a medication or a behavior therapy for alcohol use disorder, just remember that there is this safety issue of going from severe alcohol use to 0. It causes people to seize and sometimes die. Okay. Question 2, revisited. So you start developing scaledium for methamphetamine use disorder, and you learn that scaledium has been used for centuries by shaman, healers, and communities in South Africa due to its mild psychoactive properties, which that is true. Since this compound has been around for a long time, you decide to skip preclinical and safety in humans work with the compound given its history. Do you need to conduct the early safety work with this compound? Yes, no, or it depends. What do you think?
We have yes. We have 3 yeses.
Yes, you do want to do that. Awesome. Okay, one of the reasons is because it may be safe in communities out there who don't use methamphetamine regularly, but we don't know what would happen if you brought this on board and then somebody dosed really high with methamphetamine. You kind of want to know before you move to a treatment the most obvious ill effects of the interaction of the drug of abuse plus your drug that you're treating. So yes, you would still need to conduct the early safety work. It's a little frustrating, and it may actually interfere with the rapid development of medications for treating addiction use disorders, substance use disorders, but it's actually pretty important because we don't know what's out there as you bring new compounds onto people and how they would interact with these drugs of abuse that people use.
And Dr. Chaptal, we have about 10 minutes left, so.
Yeah, we should. Yeah, all right. So let me just walk you through this real quickly and then we'll stop for questions. Targets and outcomes dictate design. You'll have your slide deck to go over this. This is an important piece, and maybe I'll kind of stop here because this is what it talks about, and that is the treatment works for who it works for. This is really important and really super true. Addiction is a strange chronic relapsing disease. It's a little bit like trying to quantify God in terms of understanding addiction. And especially when you're working with people who actually get better from addiction, the issues of what actually connects them to being able to meet their use goals is really kind of weird. It's just really hard to figure out because treatment does work for who it works for. That's a whole other lecture. We can get into that. We got outcomes here for opioid trials, for stimulant trials. The question about abstinence is important, but also the reduction of use is also important. Reduction of heavy drinking is certainly beneficial. Reduction of heavy methamphetamine use is beneficial. So the idea of having softer abstinence measures and being able to look at correlates of health as an outcome is important and increasingly being recognized. Okay, so then the whole thing is about decisions made up front or regretted later. You know, the idea about making sure that you stratify variables on variables you know that correlate with outcomes, and you think first about diversity, inclusion, and equity, so that you're understanding about bringing on investigators who may be members of the community, bringing the voice of the community in to advise the trial with a CAB, and then having publication and dissemination activities that are brought information to community rapidly. So I think I'll stop here. I think I've made these points. And we will go to question and answer.
Okay, thanks, Dr. Shapta. So we will move into Q&A now. You can submit your questions via the chat box or unmute to ask a question. Once you've asked your question, please just remute to avoid any background noise. And we did have one question come into the chat during the presentation in regards to question 3 that you asked regarding the withdrawal. Would transcranial magnetic stimulation decrease those symptoms if you did that?
That's a great question. That's a really super great question. The transcranial nerve stimulation is something that I'm super excited about. We have a pilot study that's starting with this with stimulant users. And the best answer to that would be probably, but I wouldn't want to test it. So that's not one. I'm usually a pretty much of a cowboy, but that's one I don't want to do. So.
Okay, and the next question, is this a risk mitigation?
Which is a risk? Well, so generally speaking, everything is a risk mitigation, right? So like building safety into trials, having the outcomes be thought of as a way of reducing risk. I think we have to get broader in terms of, you know, being able to accept that addiction is a lifelong disorder. And if we can get to think a little bit better about, you know, what is an outcome and how does it fit in people's lives and over the periods of times in their lives, that's really important. And yes, risk mitigation, I think, is where we need to move our thinking, whether it's a medication, a behavior therapy, the interaction of these, or, you know, some sort of a community response, because, you know, this is not a neat and clean world. We live in a world where people have incomplete outcome. And if we make people feel bad about having an incomplete outcome despite doing their best, it's to nobody's benefit. We can actually help people approximate outcomes that have serious, life-changing, life-enhancing outcomes.
I had a question. Talking about the agonist approach, you touched on how it's kind of characterized as like a drug for a drug, but you said that you didn't agree with that. Can you like clarify that?
I don't. Yeah, absolutely. I do not agree with that. So let's start with heroin. Heroin, to become, when one becomes addicted to heroin, their life becomes one of avoiding relapse. Every 4 to 6 hours, someone who is addicted to heroin or fentanyl has to inject a drug or smoke a drug so that they don't get sick. It's not about getting high, it's about not getting sick. So the reason why they call it a monkey on your back is because you have to get up at 2 in the morning and dose to be able to get a full night's sleep. So you have to have gone out in the evening and scored to have drugs sitting by the bed, or else you have to go out at 2 in the morning and find somebody who's up and who will sell to you. So if you bring a medication on board like methadone or buprenorphine that actually you have to dose once a day or once every other day, what you've done is you've given people, you know, that many hours to do something other than go out and find drug and use drug and recover from the drug. So that's not, in my mind, the same thing. It's not a drug for a drug. I know a lot of community groups, and this is a really deep discussion. I'm oversimplifying this, but the importance of being able to provide a medication that lets people reach productivity, having a social life, being able to go to work and pay taxes. That's not just a swapping a drug for a drug. Same sort of idea about nicotine for smoking. So if we bring in nicotine and somebody's using nicotine replacement therapy for years or even the rest of their life, they're not going to have chronic damage to their lungs and heart that happen from regular daily smoking. So it's just not it's not an equivalent parallel swap. It's a medication. That's a great question.
Any other questions? All right, if we have no other questions, thanks again for joining today's presentation. And we would ask that you take a few minutes to complete a short evaluation, which will be entered into the chat box momentarily. It may not display as a link, so please copy and paste it into your web browser if needed. Thank you again to Dr. Shoptaw for an excellent presentation.
Thank you. And I want to say a shout out to hello to Alexa. I look forward to meeting you. You've already reached out to actually get involved. I have openings, so there are some opportunities. We're looking for people who are interested in building careers in addiction science. I do all treatment work, so this would be like addiction treatment, but we have I'm looking for people who are actually ready to do a few years of work and build their careers in this area. So if you're interested, my email is somewhere in the slide deck. You certainly should have the slide deck. There's a lot of resources in there, but absolutely reach out to me. And certainly Alexa, we've got a conversation going, so I look forward to working together with you.
All right, great. Well, thank you so much, Dr. Shoptaw, and thank you so much, Bonnie, for facilitating his talk. And next we have lunch. So we will have lunch from 1 to 1:45. And so, and then afterwards, we will have a talk by Dr. Lee again. So we hope to see everybody back in 45 minutes. Thank you so much. And if you have questions or anything, I'll stick around for a few minutes.
And I just put my email in the chat, so if people want it.
Perfect.
Christina, will you be posting the evaluation credit request for the CME?
I believe Bonnie will be doing that. Bonnie, can you respond to that? Bonnie's kind of handling the CME portion.
Yeah, so I posted the link to the evaluation and credit claiming in the chat. And so if you want to claim credit, just go ahead and fill that out.
Just up above. Oh, okay, I see it. Thank you.
Yeah.
Thank you so much. And then we'll also be posting everybody's slides on the Charles Drury University website under the SART program.
practitioners, or is it for medical students that we then use also? What's the difference?
No, this is I believe Skyscape is used by all healthcare professionals, nurses, nurse practitioners, and I mean healthcare providers, the medical providers, all the providers like PAs, medical physicians, I mean doctors, and NPs. That's why they put this.
I just want to go over it again. Her name is Dr. Sands. I think she's a doctor. She's an NP. She was hired by the BRN. Now, I don't think I think she's brand new, Leo. She's very clear that nurse practitioners are to be taught as nurse practitioners. She made that very clear on the phone with me. She said, I think you totally understand that, Dean Reckman, because you've always been in nursing. I said, I absolutely do. Now, we got to be careful, everybody.
Hello, everyone. We still have 1 minute left on lunch, but we'll start bringing everyone back. Don't want to rob folks of their 1 minute. We will be starting. Like yes, we will be starting promptly at 1:45, but it is still 1:44. So yeah, Dr. Shopta, are you okay? I'm sorry, Dr. Lee, are you okay with sharing your slides?
Yeah, I've got them. I've got them.
I pulled them up
ready
too, just
to go.
in case.
Yeah.
Awesome, thank you. And then, okay, and it's 1:45 now, so we won't do a full introduction, but just for the folks who were maybe not here for the morning session, Dr. Lee is a professor of biostatistics at UCLA and Charles Drew University. He's also vice president of clinical research and development of Prolacto Bioscience, which is a company that manufactures 100% human milk nutritional products for premature infants. I thought that was really cool when I read that. So yeah, so without any further ado, Dr. Lee, if you would like to go ahead and get started.
Well, thank you, Christina. So
sure.
Good to see everyone and maybe some more people or different people. You heard me tell a couple of stories in my morning presentation, so I'll tell you just one more. This is not a story, just when I was younger, people often asked me why I became a statistician. And I remembered something a professor of mine told me many years before at UCLA, which is where I went to school. He told me the one thing about statisticians is that old statisticians never die. They just lose their significance. So anyway, so with that, let me share my screen. And hopefully you can all see that. And
yeah, I think if you do from the beginning, perfect.
There we go.
Perfect.
Okay, so the afternoon talk is something to essentially about the design and issues with respect to randomized clinical trials. And that could be a very broad topic. And I know Dr. Shopta talked briefly, or not so much briefly, but talked about this before I did, about some of the issues with respect to substance abuse trials. I wanted to specifically drill down to, and this was something that Dr. Friedman had suggested to me as well, excuse me, to drill down to power and sample size, because that's really the question everybody always asks. I mean, I could be walking down the street and somebody says, how many people do I need? And that's always an interesting question and an important one, but it requires a little bit of thought on our part. So I wanted to take you through the thinking process of how to determine how many people you need for a study. And not surprisingly, it will depend on a number of things. Now, what we're going to talk about here, and I will show you some formula, and of course, I'll just brush through those because, frankly, hardly anybody does this by hand anymore. And so I'll tell you about some software that exists and what I like to use, and whether and whether you want to consider those, but certainly hand-done calculations are something I used to do 20, 25 years ago, but we don't need to do that anymore. Okay, so the first thing you need to know in order to think about sample size is all the different things that go into your design. And these are also important issues with respect to thinking about how to design a randomized trial in the first place. First of all, randomization. Do you have randomization? And what type you use? Now, this gets pretty esoteric because some of the most appropriate ways to design a study, or as far as sample size is concerned, get into the very specifics about how you randomize. Today, I'm going to assume the simplest type of randomization, which is essentially what we call simple randomization, essentially a coin flip. So we're not going to worry about that. But if you get into more complex designs, be aware that what I'm talking about today will have to be severely modified in order to be able to come up with an appropriate number of patients. Whether you match, now we talked about that a little bit this morning, whether you do pair matching, do you simply, this is particularly true if you're doing an epidemiological study where you have cases and then you match with one or more controls. But we also do clinical trials this way, and I'm not sure how familiar everyone is with designs like randomized crossover studies, where each patient gets both treatments or sometimes even more. But what a randomized crossover ensures is that a patient goes on a treatment, whether it's intervention or control, randomly, followed for a period of time, and then after appropriately chosen washout period where the drug, let's say we're talking about a drug, is metabolized and vanishes from the body, then they cross over to the alternative treatment. So that's the ultimate matching. And so we often do studies like that. They're not the world's greatest designs, even though they may sound like a great idea, but nonetheless, like I said, it's matched, and excuse me, the sample size will be a function of that. How many study groups do you have? That should be pretty obvious. Obviously, the more study groups, the more people you have. And then one issue which I'm not going to get into here today, but because it gets a little complex, but one of the things you need to be aware of within the context of groups is, do you have a hierarchical type design? Or put it another way, do you have groups within groups within groups? Example, I talked to this morning a little bit about a smoking cessation study we did at the VA several years ago. Well, when we randomized everyone into the two groups, whether it was this new intervention, new team care type of approach versus standard of care, we knew that we couldn't do that on an individual basis. We couldn't just simply say, okay, this particular veteran, this particular subject would get the new intervention, and somebody else would get something else, because there's the potential for contamination between the groups. It's not like a drug study where everybody's going to get either orally or intravenously or whatever, ingest a drug. Here you're giving them information, and people could, excuse me, people could talk about to one another. So in this particular study, what we did was we basically randomized hospitals. We had 20 VA hospitals around the country, and we randomized hospitals, and then anyone who was in that hospital and basically getting their primary care there would be part of the group as a whole. So there wasn't any particular wouldn't be any particular matching, not matching, but contamination of the groups, because they're being seen in different places. Another example of a study like this was a study that was done several years ago to determine whether putting calories on menus made a difference, and whether it made a difference in the way people chose to order food off the menu. Again, you could have two people at the same table, one's getting a calorie determined menu and one's not. You can't do that. So you randomize restaurants as a whole. So if you walked into this restaurant, you got this kind of menu, and you walked into another restaurant, got another one, sorry. I got something stuck in my throat. So again, that's called a hierarchical design, because you're not randomizing at the subject level, you're randomizing at the institution or a level one step up in the hierarchy. So that's something to keep in mind. Measurement type, do you have quantitative versus qualitative, or are you doing something where it's time to event, which has the potential for being censored? For example, when you're doing a study, let's say with a, for example, a drug rehabilitation type study, and your endpoint is do the patients relapse? And you're following the patients for a year, for example. At the end of a year, you see that some patients didn't relapse, some patients did. In those latter groups, you know how long it took to relapse. But in those the former group of patients where you went a year, you didn't see a relapse, you don't know for sure how long it might be. And maybe it would be a long, long time, but you don't have the ability to follow the patients that way. So we have censored data, what we normally call right censored data. That induces a different type of sample size calculation. Are you repeatedly measuring people? And then, of course, are you doing adjustments for covariates, something we talked about this morning? And then finally, one very basic thing, are you doing a one versus a two-tailed hypothesis test? Because that's going to change what significance level you use in the calculations. And now you also, then after you've decided how you're designing the study with all this plethora of things that you have to consider, then you have to ask yourself, what are you trying to show? Is it an idea of just simply estimating some particular type of some particular type of prevalence or incidence rate, or is it hypothesis testing? Again, something we talked a little bit about this morning. Now, we also have an interesting thing, which is whether or not you're actually doing a superiority versus an equivalence or lack of inferiority trial. Now, most of us usually think about when we do a study, we're trying to show something is better than something else, and that's usually the case. But there are occasionally times where all we need to show is that one approach is at least as good as something else. Why is that? Because this new approach, while it may not be superior or more effective, may be cheaper, may be easier to use, and so on and so forth. So equivalence studies or lack of inferiority trials are a totally different type of calculation or a different way of looking at the calculation than if you're just trying to show this intervention is better than something else. And when we define equivalence, we need to say what we mean. When we say two things are equivalent, we don't mean they're exactly equal, because there's no such thing in the clinical world. Two drugs are never going to be exactly the same, but they're going to be within some relatively small delta that we need to define. So that's also part of the mix. So the first thing we need to think about when we think about sample size are the errors that we make in performing, let's say, a hypothesis test. We'll come back and talk about estimation a little later. So what about errors? Because the whole idea of sample size in a study, in a clinical trial where you're testing a hypothesis about one thing versus another, is that there are going to be, there's always the probability of making an error. Okay, we talked about type 1 and type 2 errors this morning. Each one has a probability. And what we're trying to do with sample size calculations is to minimize those probabilities. We can't make them zero. That's impossible. The only way we can do that is if we sampled the entire population, but we're trying to minimize them. So what about these errors? Well, we talked about type 1 error before. That's where we say that, in fact, something really works, a treatment really has an effect, an approach really has an effect when it truly doesn't. And that, we said, is the more serious type of error. We typically set that error at 5%. And so we're just going to leave that alone. 5% is literally the level of this error. And while occasionally we might set it as low as 1%, what we'll see in a minute is that the lower you make it, the larger the sample size. So 5% is, as I say, pretty much convention. It's really type 2 error that the sample size is all about, okay? Because as I say, type 1 error is a is a fixed number based on our choice of significance level. But type 2 error, or beta error is sometimes known, or the probability of making a type 2 error, otherwise known, is called beta. That is going to be a direct function of sample size. And again, remember what type 2 error is missing something that is effective. So if we've got this intervention for, this improvement on getting people to stop smoking, type 2 error represents the probability that we fail to demonstrate it if indeed it has the effect that we hypothesize it does. So we're going to see what these two types of errors are going to do. So as I say, consumer risk, type 1 error, I call it consumer risk. The world of quality, they use that terminology. And excuse me a second, let me see my, that's it. So type 1 error, we choose as small as possible, and as I say, 5% is the compromise. Type 2 error depends on two things that we need to worry about. It depends first and foremost on your sample size, which is really what we get to manipulate. In other words, we get to choose how many people we want. And it depends on what the true effect of the treatment is. In other words, does this treatment reduce smoking rates by a small amount, or does it reduce it by a large amount? And we get to decide what we think is going to happen. Of course, we have no idea what the true delta is, but we get to pick it. We simply say, oh yeah, I think right now, right now I have 20% smokers in the VA, and maybe I can reduce that to 10% after 1 year if I implement this particular smoking cessation procedure. Okay, so that's what we think delta is. Delta is 10%, 20 minus 10. Now, again, we don't know that this procedure or this method is going to work that well, but we're designing a study so that if it does work at least that well, we'll have a high probability of seeing it, or we'll have a low probability of missing it, which is what beta is. So beta is the probability of missing this delta. 1 minus beta, which is otherwise known as power. Okay, and you can see here, 1 minus beta is the probability of finding a difference when it really exists. And we basically set, either way you want to look at it, beta, we set a beta value and then determine the sample size after we've decided how big a delta we're looking for. And again, the choice of delta is rational. I'm not going to say if I have a 20% smoking rate, I'm going to reduce it to 0, because yeah, that would be great, but we got to be rational about it and realize that it's pretty hard to, completely eliminate something. So we need to be rational. And the way we decide this number is I use the following terminology. It's the minimum clinically important difference that would make me change, if I was a physician, make me change my clinical practice. The minimum clinically important difference that would cause me to change my practice. So what does that mean? So maybe 20% down to 10%, wow, that's a big deal. I'm going to use this. But 20% down to 19%, ho hum, you see. So we pick it from a practical perspective, because we can design a study to show a 1% change. We can do that, but the sample size is going to be enormous, and more importantly, no one is going to care when that study is over with. No one is going to want to get involved with changing their whole process of delivering information about how to stop smoking based on a tiny thing like that. So we call that clinical significance. So really what we're doing here is designing a study that melds clinical significance with statistical significance. In other words, we want at the end of the day show the study is statistically significant, that is, p is less than 5%, but at the same time what we see in reality is meaningful to us as researchers, as clinicians, as whatever. And that's the basis of this. So our sample, so we pick the beta error, we pick the power is the other way of looking at it. We pick our delta, and then the sample size is going to be inversely related to those things. The smaller the beta, the larger the sample size. The smaller the delta, the larger the sample size. You can kind of see this in this particular graph, where the dotted line is the null hypothesis, That's the distribution of the data, if you will, under the null hypothesis in the top graph and in the bottom graph. And the solid blue line is the truth. That is how much better things are going to be if we implement what we're looking at. And what you can see is that that essentially when you have a very small difference, then these two graphs overlap. But when you have a large difference, they don't overlap. And what that means practically is in the upper graph, we're going to need a large sample size to see that difference. In the lower graph, we're going to need a very small sample size to see this difference. So in any event, so what's the effect of N on beta error? Okay, so here we can see that if you take that, going back to this graph and reevaluate it in terms of sample size, what sample size does is it separates these two distributions. And, you know, if you're all familiar with the concept of standard error, that's really what this is reflecting. The larger the sample size, the smaller the standard error of the two data distributions, that under H0, the other one under the alternative. And you're literally separating out the distributions by increasing the sample size, thereby making it easier to find the delta you're looking for. So in order to calculate sample size, we're going to need 4 things. We're going to need our choice of alpha and beta. Well, alpha, we know what we're going to do. Beta, as I said, is usually, or maybe I didn't say, but beta, the size of our type 2 error, most people pick either 20% or 10%, or conversely power, which is 1 minus beta, of 80 or 90%. Now, again, what does that mean if you think about those numbers? It means if you have a beta of 10% or 20%, it means 10% of the time you're going to miss a significant, a meaningful effect because of simply because of chance. 20% means you're going to miss it 1 in 4 times. So it's 1 in 4 versus 1 in 10. Most people these days, particularly that do drug studies, and I'm talking about, for example, and I've been involved a little bit in some of these COVID trials. I've been helping to design a few of them. We pick power of 90% or beta of 10%, because you'd hate to do a 30,000 patient trial and then find out that the vaccine maybe really works and just didn't do enough patients. The delta, of course, we have to pick. We've talked about that. We also, if we're dealing with quantitative data, need to have an estimate of the standard deviation. In other words, look at these pictures. You basically need to know how spread out the data is, because the standard deviation factors into the standard error. It's the numerator of the standard error. So you need to have that, and oftentimes that becomes a challenge. How do I know how different people are in terms of their let's say how many cigarettes they smoke at the beginning of a study and at the end of a study? How do I know how to estimate that? Well, a lot of times we can go into the literature and see other studies that have been done with the same endpoint, or we can always use the rule of 4. Okay, and if you're not familiar with the rule of 4, you should know about it. The rule of 4 says if you can estimate the minimum and maximum of whatever it is you're looking at, let's say number of cigarettes smoked per day. Now we know the minimum is 0, obviously. The maximum, let's say no one can smoke, for argument's sake, more than 20 cigarettes per day. I'm sure that's probably not true, but let's just say that's the case. So now you have a range from 0 to 20, or the difference of 20. A very interesting, and I won't go into the reasons for it, estimate of standard deviation. Whoop. What happened there? Oh, okay. A very simple estimate of the standard deviation is to take the range, which I just told you is 20, and divide it by 4, and we get a crude estimate of standard deviation of 5. Again, a very cool and easy way to remember how to get just a spur of the moment estimate of within or subject to subject variation. And then, of course, what we're going to do is then combine all that. One of the things that we'll see in a second when we look at the formula, the effect size really turns out to be the business part of the formula, which is the ratio of delta to standard deviation with quantitative data, which is basically how much of a difference are you looking for in terms of the numbers of standard deviations. And there's various ways that people have characterized effect size. there was a book written by a man named Cohen. Some of you may have heard of it. He was a psychologist who characterized effect sizes in terms of numbers of standard deviations in a really huge, big book he wrote about 40 years ago. That basic in looking at psychology studies, in which he looked at different studies and characterized effect sizes as small, medium, and large. Okay, small studies are those that have an effect size of about 0.2 standard deviations. Medium studies are those that have medium-sized studies in terms of the effect, are those that have a standard deviation of about 0.5, and large effect sizes are ones that have an effect size of about 0.8. So you have that kind of a way of thinking about it. Basically, we try to design studies typically so that the effect size is about at least no worse than medium. The reason I say that is because if you're really looking for a large effect size, you're probably looking for something that's unrealistic. Large in the sense of, well, you rarely see studies that have that big of a difference. I'm going to have to put you on hold for a second. Sorry about that. I've got it. Somebody's ringing my doorbell in an urgent fashion. I will be right back. I apologize. Just give me 5 seconds.
Thank you.
Okay.
Thank you. We will sit tight.
Okay.
Life in the Zoom world.
I've had some electrical problems here, and the guy decides to show up in the middle of my lecture. Okay, back to it. My apologies, everyone. Let's go back to it. Okay, now, so that's what we need. Now let's take a look at some. Now you're going to go, whoa, formulas. Oh gosh, I hate this stuff. Don't worry about it. The only reason I'm showing these formulas is so you can see where all of this stuff plays a role. And as I said, we don't do this by hand anymore. What we do is we do this by program software, and I'll talk about those at the end of the lecture. First and foremost, you can see here that there is in the upper right hand corner in the box there you can see where alpha and beta play a role. Okay, so that's nice. You can also see where your standard deviation and your delta play a role. So it all comes together into this as I promised. And again, exactly what the formula is doesn't matter. One of the interesting things, and this is a good rule of thumb to remember, if you're doing something as simple as a comparison of two means, doing a t-test, for instance, essentially, and you have 80% power, 5% significance, then this equation reduces to a sample size, and this is a sample size per group, okay? This is per group, excuse me, this is total sample size, I apologize, is 21 divided by the effect size. The effect size, remember, is delta over sigma. So if you take 21 divided by the number of standard deviations you're looking for as far as your difference, square that, you have a sample size. You can probably keep this, tattoo it on your wrist if you like, but this is a quick and dirty way of coming up with a sample size under a very common set of conditions for where you're looking for a difference between two means. Now, here we get a picture graphically of where you've got to be careful. As you can see, when you pick, this is the same, basically the same formula, but now graphed out. What you can see is for what I refer to as a medium effect size here, which is about 0.4, 0.5 standard deviations. In other words, you're looking for a difference which is roughly a half a standard deviation. So if you were talking about, for instance, number of cigarettes smoked per day, and let's say on the average, currently, a group smokes 10 cigarettes a day. And so you're going to say, you're going to want to reduce that to 5, for instance. You want to cut it in half. and let's say our standard deviation was, let's say, 2. Okay, so you're looking for no, let's make a bigger number than that. That's not a good number. Let's say your standard deviation was 10, so it's a very large variation, which is probably sensible. So now what you're looking for is a delta of 5 divided by a standard deviation of 10, and that gives you an effect size of 0.5 standard deviations. And you can see that the sample sizes are in the low hundreds, so these are rational and reasonable. Now, if you, let's say, picked a much larger difference, or your standard deviation was quite small relative to your difference, you can see your study sizes are virtually 10 or even fewer, which no one's going to believe your study if you do that size study, because nobody's ever going to see those kinds of differences. On the other hand, if you start looking for tiny differences, notice how this increases exponentially. Because remember, I said that there's an inverse relationship between sample size and effect. It's actually a squared. If you go back to the previous slide, you can see that there is a square in here. And so therefore, this is an inverse square relationship, and not surprisingly, you see this kind of exponential looking curve. So another reason why we've got to be careful about what we're looking for. Now here's an example of the comparison of two, let's say, drug cessation interventions with respect to blood pressure effect. So, not drug, I mean smoking cessation. And this, let's say these are therapeutics. In other words, you know, a drug to make you stop smoking. And so here's an example where here's the results of that study. The mean blood pressure for the people that use this particular drug. You can see 133 versus 127. You see the standard deviations, and the delta is 5.4, which is pretty large. Okay, it looks pretty large. I mean a blood pressure difference, and this is systolic blood pressure. That seems, you know, clinically meaningful. Yet the p-value here is 0.46. In other words, this is not very different. Well, you can see 5.4 relative to the standard deviations, which on the average are about 17. That's less than a third of a standard deviation. It's less than a third of a standard deviation, and as a result, you had virtually no power here. Look at the size of the study: 8 in one group, 21 in the other group. So this study was clearly is what we call underpowered. There wasn't sufficient number of patients, and as a result, it's a failed study. And that, ladies and gentlemen, is the reason, primary reason why studies fail. They don't have a large enough sample size. People didn't correctly calculate the sample size. So now, if you needed to redesign this study with the same delta, about 5, and an average standard deviation of about 17, as we said, and with 80% power, in other words, to have an 80% chance of getting statistical significance, we'd need 154 subjects, about 5 times larger than the study that was originally conducted. So clearly, what they're looking for here is a small delta. And unfortunately, I used lowercase delta here, but the same idea. And this is a small to medium effect size in Cohen's terminology. And clearly, a study of 30 patients is not going to do it. Here's just a screenshot from a program that I'll mention later as well. This is an old version of a program called PASS. Now, I'm not a shareholder in this, but I'm promoting the hell out of this particular program, PASS, because it is the world's easiest program to use, and it is extremely powerful. Means it can do a whole lot of different sample size calculations. PASS stands for power and sample size. And you can just Google it and see it. And it's not a horribly expensive program if you want to own it. And all you have to do is enter the mean, what you believe to be the means for each of the 2 groups, or in a newer version, you just simply enter the delta between the 2 groups. You don't have to know exactly what the mean is in both groups. You enter the standard deviations, and normally in most of our t-tests, for instance, we assume these are the same. and then it will produce this kind of an output. It'll tell you for different sample sizes what the power is, or for different deltas what the power is. So here, for instance, the mean for group 1 was 10, for group 2 is 11. That's 1 over 2.5, or basically a 0.4 standard deviation difference. And this is for 90% power, we need 133. For 80%, we need 100. Obviously, for the lower probability of making a type 2 error, we need a smaller sample size. Here, the difference is 2 over 2.5, which is more like an 80% or a 0.8 standard deviation difference. Excuse me, yeah, 0.8 standard deviation difference. And you only need 34. Notice how dramatically the sample size went down. And then if you increased it to 3, a difference of 3 that you're looking for, a delta of 3, well, that's greater than 1 standard deviation difference, and the sample size decreased to something virtually, you know, like, wow, I can do that study. But again, how likely are you to see a greater than 1 standard deviation difference? That's something you really have to ask yourself that. Now, if you're trying to show equivalence, the only difference in the formula is basically you have to define what equivalence is, and I'll leave it at that. But otherwise, everything else is pretty much the same. And now, what about proportions? What if you're trying to look at the proportions instead of something quantitative, you're looking at something qualitative? Then what you need to know are essentially you need to know 2 things, and don't worry about all the, you know, the gobbledygook here. Worry about what you need to know. You can either state what you believe the proportions in the 2 groups. So if you're looking at a drug, you know, a drug study in which you're trying to get people to go into one program or another, and you're looking at an endpoint of the proportion of people who are clean and sober, or whatever you want to call it, at the end of a year, then P1 would be the proportion in let's say the novel intervention group. P2 would be the proportion in the control group, and that's what you'd need to know. Or you could simply say, okay, currently the proportion of people who are successful in the control, or whatever that represents, is 50%. I want to improve that by an additional 20%. So you would therefore state the delta, if you will, from P1, and then P2, of course, would then be 70%. So either way, and then you have a formula here. So interestingly enough, when you're dealing with something as simple as proportions, then the difference in terms of what you need to know is a lot easier. You need to state alpha and beta again, of course, and you just simply need to state the 2 different proportions, or the difference between the proportions versus the control. And here's an example just to illustrate. So this is again smoking cessation for drug A is expected to be 70%, for drug B 60%. In other words, let's suppose drug B is the control and drug A is the novel approach, or the new drug. So there's a 10% difference, and you want to look at a 1% significance level, a power of 90%, you run the numbers. And again, you don't have to worry about the formulas with the software that's available. And the sample size per group is 675 patients. So with proportions, the big problem is that sample size go crazy, okay? Because you're looking at something that's a yes/no variable, as opposed to something that's just simply that's like a measurement. And so with proportions, you've got to be realistic at the one hand, but on the other hand, you know, in terms of your difference, but on the other hand, you've got to recognize that sample sizes can get pretty bad. And here you can sort of see that. Here's possible, like, baseline, in other words, control group proportions on the left-hand side. Here's the difference from that, an improvement of 10%, 20%, 30%, and so on. And as you can see, the bigger the difference, the smaller the sample size. But notice that the largest sample sizes are close to the middle. So that's kind of peculiar. If you're dealing with something around 40 or 50%, then if you're looking for differences of 10% or 20%, you can see that the biggest differences are here, right around when baseline is 40 or 50%. But as you get to the ends of the range of possible, well, of course, proportions range from 10 to or 0 to 100%. You can see sample sizes get proportionally smaller. So this kind of gives you an idea. So in some respects, you're kind of hoping you have a rare event, like 10%, and you're looking for a dramatic improvement. So right now on the smoking cessation drug A, or B, I guess it was the control, you have a 10% success rate. Let's hope that our new drug improves that to 50%, and you can see that by doing that, you only need 41 people per group. And again, here's the screenshot from PASS that shows you how easy it is to do this. And here's an example, just the output, basically the same thing we've been talking about, so I'm not going to reiterate this. Now, if you have equivalence, then the equivalence factor comes into play, and again, that's all that really changes as far as the formula is concerned. Now, if you have multiple controls per group, so that if you're comparing two proportions where you want to have like an epidemiology study where you have multiple controls versus a single case, then that's going to factor in somewhere into this formula. Again, not to worry about exactly how that works, but just recognize that you can account for that too. And what's interesting about, and people often ask, well, how many controls should I add? Because it sounds like the more controls, controls are easy to come by. Cases in an epidemiological study are hard to find, but cases are but controls you can find is, those are the people that are outside of, let's say, the zone of where something's going on. You can find zillions of those. Notice that when you get to roughly about 3 or 4, the power to detect the same difference kind of levels off. So often people say there's really not a whole much point at having 3, maybe 4 controls per case, just as a way of thinking about it. And this is when you're looking for something, a difference, or not so much a difference, but a ratio of about, an odds ratio specifically of about 2, which is a fairly typical type of delta to look for. And we'll leave that one alone. Okay. Now, what happens if and here's an interesting particular situation. What if you're looking for a rare event, something that rarely happens, but you're trying to, in this type of study, estimate the probability of a rare event. For example, you're trying to estimate the frequency of an adverse event after administration of a drug. I'll give you a specific example. When I was, one of the first early work that I did was with blood and blood products, and I was working on that in the era when the AIDS epidemic hit. And so one of the questions was, if you're making a protein, and we were making certain coagulation factor proteins that came from human blood, what's the likelihood, even after you test everything and try to do everything possible, the likelihood that you transmit HIV? Because we knew, and we still know, that the HIV can be transmitted from human blood. So how do you determine the size of a study to show that this never happens, or it happens very rarely? So let's suppose what you're hoping for is 0 events in a study. So if you do a study of N people, a certain number of people, what's the upper bound of the probability of the 95% confidence band for that true probability? In other words, if you do 100 patients and none of them have developed HIV, that doesn't mean that the drug is absolutely never going to cause HIV. There's going to be a certain probability that that could still happen, but that's our 95% upper bound. How do we determine that? Well, there's a very cool, we talked about the rule of 4s before. Now we have the rule of 3s. So you're learning all kinds of interesting things today. The rule of 3s defines the sample size for a desired upper bound. So for example, in my illustration, we want to show that the 95% upper bound for the transmission of HIV, and I know this sounds pretty high, but let's suppose we wanted to show we're 95% confident that it's less than 1%. We can never show it's 0 because we would have to study everybody in the world. So we want to be 95% confident it's less than 1%. The rule of 3 say the sample size okay, that you need is 3 divided by that sample size to get a value, which in this case is less. So the theta value here in this formula is 1% or 0.01. And so therefore you do a simple calculation, it's 3 divided by n, and that will give you a sample size of 300 in order to, if you get 300 people and none of them develop that event, you have a 95% upper bound of 1%. Now, there are some other issues that you got to keep in mind with sample size beyond what we've just talked about. Maybe you know, maybe you don't know that in the world of statistical analysis of clinical trials, there's an issue of what we call intent to treat versus per protocol. What does that mean? Okay, that means that people drop out of studies. We know this. People either don't make it through the end of the study, or they refuse to participate after a while, or they miss certain visits and you have missing data. Now, we're talking about, in this situation, completely missing observations. How do you adjust your sample size? Because in an intent to treat analysis, what that means in a clinical trial is you use everyone, whether they finish or not, okay? And that's a requirement. If you're going to publish data, if you're going to submit it to FDA or whatever you're going to do with it, you have to do an intent to treat analysis. So you have to make adjustment to your sample size for what we would call the per protocol number of people, which is what we're calculating right now. The per protocol number of people, which is the people who finish the study, do everything you want, that's what we've been calculating. But we have to account for missing data. So here's what we do. We basically come up with an estimate of the proportion of people who we think are not going to complete the study. You might say to me, how do I do that? Well, you take a decent guess. So for example, if let's suppose you thought P was 0.2, so 20% of your subjects that you randomize in your study or put in your study are not going to complete the trial. So you inflate the sample size that we just talked about, that we use these formula to calculate, you inflate it by 1, that sample size divided by 1 minus that proportion. So you divide your sample size by 0.8 if P is 0.2. So that's going to increase your sample size by 25%. So oftentimes when you're writing up your grants or your studies and thinking about what you're supposed to do, people like, oh, well, I don't think I can inflate my sample size that much, so I'm praying and hoping that I have maybe a 10% dropout rate. Okay, well, that's a lot easier, obviously, in terms of an inflation factor, but hopefully you're not far off. But this is a realistic calculation and something that you always have to consider in doing sample size calculations. Now, what about the software? What exists? Well, I've told you about PASS. PASS 2020 is the program that and it's obviously the latest version, I recommend. There's another program that's very good. I've not used it, but a lot of people like it, called nQuery. And then there are internet calculators. You can find this if you just Google a sample size for, say, comparing two means, something that, you know, we talked about to begin the lecture, using a t-test, for instance. You can use those, but use those at your risk. You don't know how well those are validated. The two programs that I'm talking about here, nQuery and PASS, have all been very well validated, and Curate, or whatever the right word is, and FDA and others certainly accept those. Now, the last thing I want to talk about is, what if you're just estimating stuff? What if you're just trying to estimate the mean and proportion of, or the mean or the proportion of a population? So, for example, let's take the proportion. We're trying to estimate, let's take an election. I guess there's one coming up, isn't there? That are going to vote for a particular candidate. How do the pollsters figure out the sample they need? You always see, we sampled 672 people, or we sampled 1,100 people. How do they decide this? Okay. So basically, what we try to do is we try to, first of all, come up with the value for the proportion itself. Obviously, we don't know it. We just guess at it. In other words, what are we going to see in the sample? You might say, okay, well, I think candidate A is going to get 45% of the vote, for argument's sake. The next thing you need to calculate, and this slide is a little, I borrowed this slide from someone else. It's not really accuracy, it's precision. The precision of your estimate. How wrong do you feel, or not so much wrong, but how much variability do you want to have about that? Because I think you all know from your basic statistics classes that what you're going to calculate here is a confidence interval, a 95% confidence interval. And that confidence interval is going to, of course, range from there's going to be a plus or minus. So you're going to calculate your P, and there's going to be a plus or minus from about P, okay? So P plus or minus this precision that we're calling D here. You decide what you want D to be. And by the way, what people call D is margin of error. So when you see margin of error, it's just basically selected. That's why you see the same numbers over and over again. 3%, 4%, 5%, 2%, whatever it is, that's the margin of error. And so you put that margin of error in there with where you expect the proportion to be. And by the way, most people just assume P is 0.5. Okay, the reason they do that is because that maximizes the sample size. In other words, it's the biggest sample size you need to estimate the proportion who are going to vote for candidate A with 95% confidence. And then finally, this Z value, which is from the normal distribution, it's the Z value that you need for 95% confidence. It's the same thing is true on the other side for estimating a mean. The only difference is instead of proportions, we're dealing with standard deviations, and we're dealing with, again, the margin of error, which you're putting on, say, the estimated, say, mean number of people. Let's suppose you were trying to figure out how many people are going to vote on November 3rd. All right. Well, you know, you don't have to know how many. In this case, that's unimportant. What you do need to say is, what kind of variation do you expect to would you expect to see in the sample? In other words, if you took a sample of people and asked them, are they going to, you know, how many are going to vote? Try to get how many are going to vote. There's going to be a variation in that from sample to sample, and so that's going to come into play here. The precision, the plus or minus on your estimate, and obviously again your confidence level, which is what Z reflects. So here's a couple of examples just to illustrate. So on the one side, on the quantitative side, we want to estimate the average number of weekly drinks consumed in a VA population. How many drinks does the average vet take? And we want the error of estimation to be within plus or minus 2 of the true mean. So we want that to be our margin of error. And we suppose that the standard deviation, in other words, the variation of number of drinks from person to person, is 5. You put that all in, you only need 24 people to accomplish that, okay? Now, another example of using proportions, the expected recidivism after treatment for cocaine addiction is around 20%. We want to estimate the true prevalence in a population within plus or minus 5%. So you're supposing it's 20%, but you want to see what it really is by going out and taking a sample. Notice here, to get to within plus or minus 5% of the truth, you're going to need 246 people to sample. So proportions, pound for pound, are always going to result in bigger sample sizes. So what we often tell people is if you can avoid, if you don't have to estimate something that's a yes/no variable, try not to, because you're just going to be stuck with a much larger sample size than if you were trying to estimate some quantity on these people. And that, ladies and gentlemen, is the end of the lecture. So I hope that, let me get rid of this and get back to seeing you all. And I hope that provided you with a little edification on how sample size works, albeit in very simplistic terms or situations, but there you have it, in less than an hour.
Great job, Martin. Thank you so much.
Okay, Ted, no problem.
Questions from anybody? I think there was a question in the chat.
Oh, okay, let's take a look at chat. Okay. All right. Oh, a couple of LOLs. That's good. Thank you. I appreciate that. Do you recommend using G-Power software? I must admit, Anna, I am not familiar with that program. I'm not suggesting that there aren't other programs other than the two that I mentioned. I think from people's perspectives, if it's easy to use, and if it's been around a little while, look for comments on the, obviously on the internet for things. That's what you're looking for. I can tell you at the very least, again, I'm promoting PASS, but that's because I love it so much, that it is extremely easy to use. And that's what I find most attractive in any software. And I'm sure most of you agree with me.
Does anybody else have any other questions for Dr. Lee?
Okay, well, thank you, Anna. I appreciate that.
Thank you, Martin.
Just a comment for Dr. Lee. You made it very interesting and not boring, so I really enjoyed you.
Thank you, Angela.
Thank you. You're welcome.
All right. All right, everyone have a good rest of the, I know there's a couple more lectures, and have a good weekend.
Thank you, Martin. And we'll pick it up at 2:45.
Yep, that sounds great. Yep, at 2:45 we have Dr. Harawa up next. Yeah. So we have a 5-minute break. So if everybody wants a chat break, a stretch break or snack break or even a chat break, that works too. Feel free to take one. And Dr. Harawa, I'm going to make you a co-host so that you can share your screen. Are you okay with that?
Yeah, that sounds good. Thank you.
Great. You're welcome. Okay, wonderful. Oh, thank you. I just saw your message in the chat. Dr. Hurawa, we're ready for you whenever you are. And Dr. Friedman, if you'd like to introduce her as well.
Hello, sorry about that. Hi.
No worries.
Okay, are we ready to start with Dr. Rawa?
Yep, we are ready.
Okay. Dr. Rawa is also a very close colleague of mine. She's and I've known her for many years and work very well with her. Dr. Rawa is an associate professor with the Charles Drew University of Medicine and Science and an adjunct associate professor at UCLA. She's trained in epidemiology. Her research involves understanding trends in HIV and other sexually transmitted infections and developing effective, culturally relevant prevention interventions. She has partnered with several local health organizations to conduct innovative, community-based research in a variety of populations, including high-risk African-American men, sexually active African-American and Latino women, older adults, and incarcerated and post-incarcerated men and women. In order to foster methodologically sound and effective approaches to address health disparities, Dr. Ra has also written about conceptual issues and the categorization and analysis of racial and ethnic groups in healthcare. She regularly shares her knowledge and findings by presenting data to community policy and academic groups. In addition, she launched the Breaking the Silence events, a series of one-day conferences and trainings designed to educate and empower women of color and their providers in order to improve sexual health. Dr. Harala has received numerous awards and has a current R01 grant and also received the Unsung Hero Award from the NAACP. Nina, thank you for joining us today, and your contributions are appreciated.
You're welcome. Good afternoon, everybody. I know it's afternoon after a long day of, I'm sure, really stimulating discussions. My focus today was going to be on both research and recruitment and interviewing. I decided to focus in on interviewing, and I can spend a little bit of time in the questions and answers and at the end talking about recruitment. But we're certainly in a different situation now in terms of how we recruit people for substance use research or any other types of research because of COVID. And so, you know, I think I'm in some ways I'm learning in that context as well. And I thought that there's certainly a lot we could speak about in terms of interviewing techniques. So I wanted to focus there. Thank you for that introduction, Dr. Friedman. I realize it's actually a little bit old, so I technically am a professor at this point, and I also want to, full professor at this point, and I also want to thank Kiana Montezeri, from whom I adapted a lot of these slides. And I'll just say a disclaimer in a way, which is that a lot of the slides were developed for a study in which we were training our interviewers on a study focused on African-American men. And so that's kind of the imagery that you'll see in a lot of the slides. But I realize that our group is more diverse, both in terms of race, ethnicity, and gender. I just decided not to focus in on modifying all the pictures for today. So my goal for today is to review with everyone what is survey research, your role as an interviewer, and then standards and rules for good interviewing. And, you know, so to start with the big picture and then really hone down to some of the specifics. And if people, are we just doing questions at the end, or can people do them along the way? Does it matter?
People could do them along the way, or you could take a break and ask questions if you want.
Okay.
So we like to involve the audience.
Good. And I would like that too. So I don't always see, I find when I screen share, I don't always see things pop up in the chat, but I'll try to stop along the way, or if somebody needs to yell and get my attention, they can. So let's start with the big picture. What is survey research? What does it mean in the first place? So survey research is a systematic process in which we're gathering information from individuals in order to answer research questions. And it involves some key tools, an important one being the instrument, which is the questionnaire used to collect the data. And that's the instrument that you would be using to collect data from individuals. There's the person collecting the data, the interviewer. And although a lot of data sometimes now are collected, without an interviewer per se, but since this is specifically, designed for teaching people how to do, do interview-based research, there's of course a data collector. The people providing the information, the respondents or the participants, the people who are sharing with us their information, sharing with us their data. And then the participants as a whole provide the sample, the group from which we're going to make conclusions and make inferences to a larger group. Now, I realize that a lot of you are not necessarily, I realize that survey research can come in many different forms. It's not always going to be community-based research, but CDU has a pretty strong history of community-based research, and a lot of the key thinkers doing community-based research, are at CDU or have been at CDU. So I just wanted to include this definition of one of the more intensive forms of community-based research, community-based participatory research, which is a partnership approach to research that equitably involves, for example, community members, organizational representatives, and researchers. And those individuals or those entities are involved in all aspects of the research process. And all of these partners contribute both expertise and have shares in the decision-making and the ownership of the data that resulting data. And I partly wanted to just put this out there in the sense that when you're thinking about the specific research study that you're a part of, I think it's useful to understand as an interviewer, as a research assistant, or whatever your role might be in a project, the extent to which the work is actually informed by the community. Because I think that helps you to feel confidence in the work that you're doing, the more that it is informed by the community, and perhaps respond to questions that you might be getting from study participants. So I want to now talk generally about what's expected of a good interviewer. So there's, kind of four main aspects of this. You have to be able to collect the data and do so in a manner that's both efficient and with high quality. You have to be able to work well with people and be both professional and friendly. In other words, be able to make a connection with individuals from which you're collecting data, but not necessarily in a way that's so casual that people sort of forget the purpose for which they might be sharing their information with you. Almost always you're going to be doing this work in a team, so being able to work well with others, to communicate well with others, and to recognize that it's not about any one person, but about the team working efficiently together. That's really key. Being persistent. I think doing both interviewing and recruitment for studies often requires a great deal of perseverance and a pretty tough skin. So I said I wouldn't talk a lot about recruitment, but I will maybe spend a few moments here because in our various studies whether one might be recruiting people from a clinic or like from a clinic waiting room or online or doing street-based recruitment, any of those types of recruitment, it involves a great deal of rejection oftentimes. A lot of people will, walk by without even looking at you. Other people, will look at you but not, literally pretend that you're not speaking to them. There may be any number of responses that you get from individuals. And so it's really important that you're willing to kind of just realize that that's part of the job and not take it personally, and still be able to approach the next person with a smile, or the next person with whatever kind of positive energy might help attract them and encourage them to be part of the research that you're part of. The other reason in this particular example, we also talk about keep trying until respondents turn up. And a lot of the work that we do, working with people who may experience instability in their housing, may experience involvement in the criminal justice system, may not be able to keep their cell phone and pay their bill off every month, and so their numbers change, or they, for a while nobody can reach them. So that requires, if you're going to be doing research in which you're following people over time, there's a great deal of perseverance there too, and you have to be willing to keep trying until you do find somebody. And in our experience, I've had plenty of times where we literally thought somebody would never, we've tried 15, 20 times to reach somebody different ways, and 2 months later, they're calling us. And for whatever reason, in that period when we were trying to reach them, they couldn't respond, but our perseverance, inevitably, at least at some level in the future, paid off. And finally, a good interviewer is going to be reliable, meaning that they will, I already talked some about communication, but they're going to show up when they're supposed to. It's fine. It's not fun, but it's fine for our participants to be late and not show up. As an interviewer, we can't do that, right? Our staff members can't, have an interview with a person and then not show up and not let them know, or show up late. The reliability on our team is really, really key. And one of the things that we've also found through doing this work is that it's very easy to get a bad reputation in the community. If you have some member of the team who maybe makes people feel uncomfortable or isn't reliable, and then you start having people in the community saying, don't go, don't show up for that study. They I had this bad experience. That can make it very difficult to recruit people in the future. Good interviewers keep in mind their responsibilities. A key piece of that is keeping track of your belongings, your paperwork, your electronic files, right? All of those things that are related to the study. And some of this is a little bit more focused on field-based research, but it's really important that people are aware of all those things. And I think having checklists before one starts an interview, checklists at the end to make sure all of those little things that one has to remember are taken care of, can be really, really helpful. Responsible for checking your phone and your emails to ensure that communication is working appropriately with your supervisor and the members of your team. With maintaining your own personal safety while working in the field, and probably there are levels of this as well, even as people are doing interviews online or in other contexts, but maintaining both one's own personal safety, but that of the people that they are interviewing. And I could give an example of this. This has both to do with safety and privacy. We were doing a couple years ago a study with African-American women whose partners might have increased risk for HIV. And one particular day, a woman came with her partner, and he was waiting for her in the waiting room while she was doing an interview. And he came up and asked me, or asked somebody else on he asked me and somebody else on the team, so what is she doing in there? Is she smoking in there? And I was like, wait, what? He was like, well, she told me she's doing a smoking study. Is she smoking in there? Are they having her smoke? And we weren't doing a smoking study. Dr. Friedman does smoking studies. And we probably wouldn't have people smoke if we were, but I had to just say, no, she's just doing an interview. We don't have people smoke for the study. But that's what she had told him, right? That the study was about smoking. And so I couldn't, I was not going to, he knew she was there for a reason, but I couldn't give up anymore. And I had to be we had to be quick on our feet to think about, something to say that wouldn't make it seem like she was lying, because for whatever reason, she didn't tell him the full truth of what the study was about, but also not give him any more information that he needed to have. And part of why I put that under safety is because I'm not saying this was true for her, but there were certainly many women in this study who talked about domestic violence as being part of their experience when we asked questions about intimate partner violence. And so, we had to be very careful of that. Even when we were reaching out to contact people for follow-up interviews, we didn't say anything that somehow raise suspicion or concern in their partners in case they were in a relationship where that safety wasn't there. So, you know, related to that, of course, we always have to maintain the confidentiality of participants' data. And that really means, you know, following all the different rules that are set out within the city guidelines and by the institutional review boards. I think because of the ways that our cell phones and our laptops and everything is now connected, there's both enhanced security, but I think also enhanced risk to that. And it could be as much as, say you didn't close something out and you give your phone to somebody else and they somehow see something about a person on a file that you had open on your phone. As a potential example. So really setting up your screen savers so that they turn on and have passwords, ensuring that you're only working on secure devices, closing those things out, using appropriate passwords. All of those things are key. And because, you know, unfortunately having had the experience of having staff who we've had a laptop that was used for data collection get stolen. We've had recorders that went missing. They actually weren't stolen, but took a long time to find. So, you know, all those things having to report to the IRB is not a fun experience. And of course, you never want to have a situation where data that somebody reports to you in a study later leads to legal problems or problems with their employment or their partners. The last thing I have on here is the importance of speaking up. You know, as a researcher doing work with a range of different communities, and Dr. Friedman talked about some of the different communities that I've worked with in the past, you know, I do my best. You know, sometimes I might be closer to the community than others, but a lot of times I'm not necessarily part of the specific subgroup or community that we're doing research on. And I do my best to learn as much as I can about them, but my staff are often closer. And so I really feel like it's key that my staff, if I'm asking, say we have a survey instrument and we're asking about something in a way that participants really wouldn't understand, the language used is not appropriate to how participants themselves understand or talk about something, I really want my staff to speak up. I think it's really important that when they notice something because of their own experience and background or membership in a community, that they speak up, and I'm always willing to listen and hear what they have to say, because no one person, no one leader, no matter how many degrees we have behind our name or experience, is going to know everything they need to know to always be able to capture information in the best way from a particular population. So sorry, I've talked longer than I planned to without stopping for questions, but does anybody have any questions or comments before I keep going? Unfortunately, I think I put Miss Plus to sleep, so. No, okay, I didn't. Anybody have any comments?
You have a soothing voice.
Thank you.
Yeah.
Hi, Robert. I'm sorry, I'm cheesy, Miss Plus.
Sorry, I just want to really reiterate, I've been part of projects where people that were in charge of research didn't really see the interviewers as professionals or as people that could extend that professional aspect of the study to the research participants. And so just the last comment that you said, I thought was really valuable and important, especially when talking about research that deals with invisible populations or vulnerable populations.
Yeah, that I mean, it's unfortunate that I think sometimes researchers don't value that expertise in their staff, but I think that there are a lot of researchers who do, and I'm certainly one who does. And, you know, I would hate for you to tell me 2 years later, like, why were you calling, you know, nobody talks about it this way, I always talk about it this way. You know, tell me that early on. That's stuff we can change and address. So thank you.
And this is Plus again. Thank you, Nina, for that presentation.
I'm not done yet, but go ahead.
You know, I've been on here since 8 o'clock this morning, but and 2 of my mentees are on the call. But you know, I'm glad you said what you said, because you know, a lot of times the research, you know, I mentioned earlier this morning about the community faculty at Drew. And so a lot of times the researchers don't value what we know of life experience with their research. And when you just said, you know, they see it one way, or whomever says one way, they hadn't thought about it that way. But then when you bring in a person that's lived the experience that you're trying to do, they come from a whole different. I've heard some technology, some words today, and I'm like, this is not community friendly, you know. But I went along with it. But like you said, you know, we might bring it our view, and if you're willing and you're and we know you're able, but if you're willing to open your mind to say, oh wow, I never looked at it like that, and it could be so simple and so instead of so technical, you know what I'm saying?
Mm-hmm.
And you respect what I'm saying, and then we give value to the community person that's speaking, and then that's how you get the trust factor in there also.
Mm-hmm. Yep. Thank you. So I'm not going to go down to some more of the specifics, and you know, kind of as an overview, the key to how you're involved as an interviewer is both following the steps as they're laid out in the study protocol, and doing your best to both remain bias-free, although you know we're all human, so at some level sometimes our biases and our judgments come out, but at least to maintain as much as possible the appearance of being bias-free. So I'm going to talk both about your role as an interviewer and working by the rules. So even though I'm going to go by this quicker, you'll see all these again in a minute. So as I said, an interviewer needs to be both professional and friendly. We want to gain the cooperation of the person that we're working with so that they want to, they not only are willing to participate, but they're willing to do what it takes to finish the survey. That may mean going through, I tend to have long surveys, so that may mean 200-300 questions actually, which is, you know, about an hour or so of questions. Being willing to be honest, being willing to listen to the whole question, right? Even if they might want to move through faster. Building a rapport so the individual feels comfortable with you. But at the same time, being professional so that they don't necessarily feel like that they're there just to tell you about everything about their lives, or that they're there to hit on you, which sometimes happens with participants, or they're there to, you know, any number of other things. So it's that kind of line where people feel comfortable with you as an interviewer, but they also know they're there to for a specific purpose, and that you're there to help fulfill that purpose. So the next kind of piece of that is being neutral, and I would really sort of put that in the lines of both neutral and non-judgmental, that we work with individuals, for working with people who are involved in substance use disorder, involved in substance use. They may be involved in activities that are illegal. They may have substance use disorders, which is often a stigmatized condition. They may be involved in other types of activities that are illegal, or be members of groups that are stigmatized. So all of those things means that the person in front of us is used to, or could be used to, being very much judged by society. And the last thing that we want is that fear of judgment to shift what they might, the information that they might give us. So we want to ask the questions that are on the survey and respond to questions that they might have in a way that doesn't seem to sway them in one way or another in terms of how they respond. I already talked about maintaining confidentiality. Another key part is to record information as accurately as possible. So really having that attention to detail and knowing well the instrument ahead of time, so you know as an interviewer what it is that the questions are trying to get. The key words to survey research are what we call standardization and neutrality, that people are ultimately asked the questions in a similar way as possible. And that, well, I already talked about neutrality, that there's nothing that might push them in one way or another kind of away from the truth in terms of their responses. So those are key pieces to what I'm talking about. Related to the issue of neutrality, it's really important to think about the language that we use. And I think this is another place that sometimes as an interviewer, you might end up speaking up in terms of language that's used in a survey instrument, or that are used otherwise in the work that might be done. But I shared this specific sort of discussion, especially, and this focuses in on addictions and really tries to push people away from language that's considered stigmatizing. And I have to, say very honestly that I think this is a very much of an evolving landscape that's happening. We've evolved as a community in terms of health towards much more person-centered language. And within substance use, we've moved away from certain types of terms that were very common within substance use. And I have family members who are part of recovery communities, and in those communities, people use terms like addict, right, to refer to themselves. But as researchers, those researchers or providers, it may be problematic for us to be able to using those terms to refer to people, because in part because it focuses on the disease and not the individual. So you can see on this list here, they encourage people to say person with a substance use disorder as opposed to addict, or much more problematic terms like junkie or druggie, to avoid terms like drug offender. And there's a whole slew of words associated with people involved with criminal justice that can be very problematic, that tend to be used often: ex-offender, convict, felony involved. A lot of these things just really focus so much not on the person, but on the thing that they might have been criminalized or for. And so you can see here, there's a list. I've got a few comments in the chat, so I'm going to read those, but then if somebody has some other examples, I'd love it if somebody could also bring up some other examples of common language, but that's ultimately stigmatizing. So in terms of comments here, so some people are saying they can't see this. I don't know that I can enlarge it. I have it in the fullest screen view. So I'm sorry, a couple people are saying they're having trouble seeing this. So I'm going to read some of them off. Ms. Plus and Karen, can you tell me, are these all hard to read, or is it this particular one because the font on this is small?
The font on this one seems a little smaller than the rest, but I didn't have, personally, I didn't have any issues reading the other ones, but.
No, the other ones are fine. It's just this one.
Mm-hmm.
Okay, great. So yeah, that's all. I can read through some more of these. And then Robert asks, how do you balance remaining neutral to collect clean data and giving them information and resources which might help the situation? Okay, I want to come back to that because I think that's another tricky one. So I'm just going to go through this list because it's not long and I think it's useful. So they encourage people to say person living in recovery rather than ex-addict, person living with an addiction rather than battling or suffering from an addiction, a person arrested for a drug violation rather than drug offender. They also encourage people away from terms like non-compliant or bombed out, and to say something like chooses not to at this point. Not using medication as a crutch, instead medication as a treatment tool. We have some really powerful medication assisted therapies, but in some communities, those are stigmatized, including some recovery communities, those are stigmatized. And there are people for whom, medication assisted therapy is very much a lifesaver. And so, these terms like medication as a crutch can be really problematic. They suggest against relapse. To be honest, I haven't gotten rid of relapse as a term, but instead they had a setback. And then, I think these last two I think are common, but obviously kind of problematic, saying stayed clean, because the opposite of clean, right, is dirty, or a dirty drug screen. They, describe both of those as potentially stigmatizing and suggest instead of stayed clean that you say maintained recovery. Instead of dirty drug screen, say a positive drug screen. So any thoughts on these? So going back to Robert's question, so he was asking, how do you balance neutrality with providing resources?
And I think
yeah, I'm sorry, you had a question?
Yeah, Juanita did.
Can you hear me?
Sort of. Go ahead, Juanita. No, we can't hear you. I'm sorry, I can't hear you, Juanita. Type it in.
Type it in the chat, maybe.
So then I'm going to go back to Robert's question, which is maintaining neutrality and providing resources. So I think the best way to do that is to to separate the two processes. So, on a survey, people might be asking for resources, or perhaps they're showing a need for resources. And depending on your particular study, it may well be appropriate to do that, but that could be something that's done at the end and not along the way. I will say that the model that we've often used is to let people know that we provide resources and referrals, but not necessarily giving them unless they ask, with some exceptions. So that if, like in the study when we were asking about interpersonal violence, we specifically offered resources for that in case somebody was in a dangerous situation. And we had a kind of a computer assisted interview. So there was literally a flag reminder at the end for the interviewer to get some more information and, offer both immediate and longer term resources for that. And then also when we do depression screening, if somebody endorses like suicidality in their questions, then we always offer something regardless if they ask, because then those are situations where there's potential for immediate danger in terms of individuals. And I think if somebody, say, maybe doesn't ask, but they start going on, going off the survey to go on about a problem, then I think it's fine to redirect and say, if you would like services about this, can we spend some time at the end? I'd like to get through the survey and to provide that at the end. Now, if they're distressed, though, I will just add one more caveat. If they're really distressed, like you really feel like this is the questions that you're asking in a way is kind of causing them distress, then it's also might be appropriate just to give them a break or to stop and continue at another time, because we don't want our questions to cause harm. And I think they have some potential to do that. For, when we ask a lot of questions about traumatizing events, sometimes people need to stop and be able to regroup or talk to somebody in that. So, I can't say we've had that a lot. We've always trained our interviewers to also try to detect those things. I think there were, let's see, one or two more questions. Oh, so you suggested homelessness versus person dealing with homelessness. I also, sometimes now people also get away from homeless. They say houseless or shelterless, right? Because oftentimes people have a home. They might have a place they go to regularly. It just may not have a roof over their head. And then Karen asked, are interviewers mandated reporters? So my understanding of this is, essentially, yes, that we are expected to report if somebody reports abuse to a dependent, so generally to a child or a dependent elder adult. The reason I'm just slightly, well, yeah. So, but both your PI and the consent form should detail that. If there's any questions that might get to it, they should really tell participants ahead of time that this is something that you're expected to do. The limits to confidentiality should always be spelled out in a consent form, because then a participant, if they really don't want you going down that path of reporting, they can choose not to tell you something. Okay, I'm gonna keep going. So as I said, one of the keys to being a good interviewer is working by the rules. They may not always be fun, but the rules really help maintain the quality of the study, the information that's there. They avoid bias, which can creep in different ways. I think I've already talked to you about some. But it's also important to realize that it's not always easy. Oftentimes our participants are vulnerable. They have a lot of needs, and so there might even be an emotional toll on us as they're telling us and in responding to these questions, along with maybe the emotional toll that they might experience. Sometimes participants don't act in a way that's appropriate, and that's one of the reasons. The more professional we are, I think the more we signal to them what's appropriate and what's not. But they may not always act appropriate. And of course, your safety in that is important too. If somebody ever acts really inappropriate, then you need to maintain your own safety as well. Participants can be needy or really need our services, and started to talk about that. But it's also really key to remember that participants are our community, that they're part of the community that at CDU we endeavor to serve. They're why our institution was built. And I really have always liked the term participants, and I always try to get people to think like, you're part of the study, you have this key role, we need your information, your life experience is key to the success of what we're doing, and your being part of it has a potential to help people down the line. All of those are things that I'm always asking that my team members are conveying to people in our research, so that they really are participants, not just subjects. So getting into some of the specifics of the rules. So maintaining neutrality, and part of this is not suggesting an answer. So I already talked about a judgment, but sometimes it can be easy, very human, to suggest an answer, to assume you think what somebody means, what somebody means when they don't answer directly. And we want to avoid that. I'll give some specific examples. Don't explain questions unless you already have a detailed probe in terms of how you're supposed to explain it. Do not give your opinions. So like if somebody makes a comment, okay, thank you. You don't necessarily want to assume, great, oh, you did that. Again, staying away from the sense that some way somebody might perceive you think their response is good or bad. Key to maintaining privacy is also not discussing anything that somebody told you with people outside of the project. And one of the challenges sometimes, in my work, that we've sometimes had team members who end up knowing somebody who's in the project. This goes to my second bullet. And so they it would be especially problematic, right, if they started talking about what a study participant said, because they're part of the same social network. So if you are ever in that situation where you know somebody who's in the study or are in some other way connected to somebody in the study, say they're your sister's boyfriend's friend or something, it's important to talk to a supervisor about that, so that they can put in place whatever protection might be needed, so you avoid a situation that where the person maybe might feel rightly or wrongly that you shared information you shouldn't have. And then if a person is in rehab or receiving services somewhere, and say you're also working with those individuals who work there to recruit other participants or in some other way, it becomes extra important that you don't reveal their responses, right? You might be at a place where people have to be sober to stay there, and a participant might tell you that they went out or they managed to sneak in some kind of substances last week or the day before. Even though that's not allowed and it's against the rules, you can't share that information, right? The participant told you this in confidence. And so in any of those types of settings, it's really important to maintain confidentiality. I have a staff or team member who works on a study and has done a lot of work in Cook County, Chicago, in their jail. And one of the things that she told me is that even though she works well with like the deputies and the custody assistants in that setting, that after she interviews somebody, she's very careful not to spend any time like talking or joking around with the deputies, even though she has a good rapport with them, because the last thing she wants them to think, the person she just interviewed, is that she's over there joking with the deputies about something they just said or telling them. Even though she would never do that, it's also just avoiding that appearance, right? That this is what the subject of the conversation is now. So this goes to some of the specifics. It's really important you read the questions exactly as they're worded in the questionnaire. So that's the standardization. Everybody gets the same question. To do this well, you need to be familiar with the questionnaire. And part of that becoming familiar is really knowing what they're asking for. In case the respondent has a question, you should know what was really being asked for. But ideally, the interview should have specific probes or specific responses. So if somebody asks, well, what do they mean by this? I established ahead of time should have been the way you can respond to those types of questions. You should be able to dig deeper for additional information if that's needed to fully answer the question. And kind of like I said at the bottom, you really want the respondents to interpret the question as they see fit, unless you're told a specific way to explain a question in the training for whatever interview you're doing. Because if and the reason for this is if I explain a question one way and Robert explains it another way and Ms. Pless explains it another way, then everybody's not getting the same questionnaire, right? So that all needs to be as consistent as possible. So I'm going to skip through some of these. I'm afraid I'm already getting closer on time than I thought. You know, I would say relax. Even long interviews, we've often find that people kind of enjoy it. I think oftentimes people are not asked a lot about themselves or their lives, and it actually makes you feel important when somebody spends an hour, half an hour with you asking questions and listening to your responses and really focus on you. So you could, I think you could often expect that it actually may not be a chore, but something that people at some level enjoy. We encourage you not to accept a don't know. Generally, don't know can be a lazy response. Oftentimes you can get people closer to a response than a don't know. Not always, but often you can. You always want to thank people for participating. Make eye contact unless you really get the sense that they're not comfortable with it. It's also important to learn the lingo, and I specifically pulled out these two slides because it made me laugh that they were so different. So, in any substance use survey, they're going to have the names of the drugs, but then they should ideally have what the more common street names for them are. And so I literally went on the online last night and I found this list of names for ecstasy. And can anybody tell me something strange about these two lists of these nicknames?
Well, the writing is very small, so I can't see it.
Oh yeah, you can't, sorry, people can't see them again. So they're almost completely different. So, I mean, part of my point is I think it's important to know the lingo, but you also have to realize that the lingo changes and the lingo differs from place to place. That ideally, if you are doing community-based research and you have partners at the beginning who can help tell people what the lingo is. And then you might learn things along the way. Somebody, you ask about ecstasy or X or E, and then somebody says, do you mean beans or clarity? And then hopefully you can maybe have a little conversation with them to make sure you're talking about the same thing. But there's going to be a lot of different names for different substances, or sometimes for them when they're combined. And so I think it's important to get this information, but to realize that it's very time and place specific, and sometimes community specific, right? Different ethnic groups, different subgroups might call things differently, even in the same time and place. So as you're going through the interview, part of the professional part and part of what you're teaching the respondent is to get used to you reading the whole question because they often will want to answer before you're done, and to answering only based on the responses that are available for that question, because people will give you a response and then you're trying to figure out, well, which one is it? And so instead of you making that choice, you want to get into the habit of telling them what the responses are, or showing them what the responses are, and knowing that they need to pick one of the responses, not you. You always want to ensure that the participant knows the time frame that you're asking about. And in some surveys, this might switch around. Sometimes you're asking about the last 30 days, sometimes it's the last 90 or a lifetime. So whenever it switches, I always try to stop and make sure somebody knows, okay, the next set of questions are about the last 30 days. That might be, so that's from today, October 15th to, back to September 15th. And really so that they're clear what's the time frame. So I'm going to go through some more specifics. Oftentimes, the responses we give people are very simple. A, B, or C, and they might have very simple, but the respondent might not answer that simply. So you want to encourage them to respond in a way that fits the responses. You want to give them time to think about their response. And sometimes they might struggle or not understand right away, but just repeating and giving them a little bit of time, repeating the question as it was written, and then it might click for them. And then when you do try to get more specific, it's important that you don't direct people one way or another. So somebody might say, we might ask, how often did you use cocaine in the last month?